8. The nervous system. Learning outcomes. These learning outcomes tell you what you should be able to do after completing the chapter. They correspond by number to this chapter's sections. 8 to 1 describe the anatomical and functional divisions of the nervous system. 8 to 2 distinguish between neurons and neuroglia on the basis of structure and function. 8 to 3 describe the events involved in the generation and propagation of an action potential. 8 to 4 describe the structure of a synapse and explain the process of nerve impulse transmission at a synapse. 8 to 5 describe the 3 meningeal layers that surround the central nervous system. 8 to 6 discuss the roles of gray matter and white matter in the spinal cord. 8 to 7 name the major regions of the brain and describe the locations and functions of each. 8 to 8 name the cranial nerves, relate each pair of cranial nerves to its principal functions, and relate the distribution pattern of spinal nerves to the regions they innervate. 8 to 9 describe the steps in a reflex arc. 8 to 10 identify the principal sensory and motor pathways, and explain how it is possible to distinguish among sensations that originate in different areas of the body. 8 to 11 describe the structures and functions of the sympathetic and parasympathetic divisions of the autonomic nervous system. 8 to 12 summarize the effects of aging on the nervous system. 8 to 13 give examples of interactions between the nervous system and other body systems presented so far. Clinical notes. Demyelination disorders, P251. Epidural and subdural hemorrhages, P262. Spinal cord injuries, P264. Aphasia and dyslexia, P274. Seizures, P275. Cerebral palsy, P289. Alzheimer's disease, P296. Spotlights. The generation of an action potential, pages 254 to 255. Propagation of an action potential, pages 256 to 257. An introduction to the nervous system. 2 organ systems coordinate organ system activities to maintain homeostasis in response to changing environmental conditions. They are the nervous system and the endocrine system. The nervous system responds relatively swiftly but briefly to stimuli. In contrast, responses by the endocrine system develop more slowly but last much longer. We consider the endocrine system in Chapter 10. The nervous system is your most complex organ system. For example, right now you are consciously reading these words and thinking about them. At the subconscious level, outside your awareness, it is doing much more. Your nervous system is also monitoring the external environment and your internal systems and issuing commands as needed to maintain homeostasis. Yet in a few hours at mealtime or while you are sleeping, your pattern of nervous system activity will be very different. The change from one pattern of activity to another can take place in almost an instant because neural function relies on electrical events that proceed at great speed. In this chapter we examine the structure and function of the nervous system. We consider its cells through their organization into 2 major divisions, the central nervous system and the peripheral nervous system. Build your knowledge. Recall that the nervous system directs immediate responses to stimuli, as you saw in Chapter 1, an introduction to anatomy and physiology. It usually does so by coordinating the activities of other organ systems. It also provides and interprets sensory information about internal and external conditions. 7. 8 to 1 The nervous system has anatomical and functional divisions. Learning outcome. Describe the anatomical and functional divisions of the nervous system. The nervous system carries out 3 main functions. It 1. Monitors the body's internal and external environments. 2. Integrates sensory information. And 3. Coordinates voluntary and involuntary responses of many other organ systems. The nervous system has 2 major anatomical divisions. The central nervous system, CNS, consists of the brain and the spinal cord. It integrates and coordinates the processing of sensory data and the transmission of motor commands. The CNS is also the seat of higher functions, such as intelligence, memory, and emotion. All communication between the CNS and the rest of the body takes place through the peripheral nervous system, PNS, which includes all the nervous tissue outside the CNS. Figure 8-1 presents an overview of the functional relationships of the CNS and PNS. Notice that the PNS itself consists of 2 divisions. The afferent division, afferents, to bring to, of the PNS brings sensory information to the CNS from receptors in body tissues and organs. Receptors are sensory structures that either detect changes in the environment, internal or external, or respond to specific stimuli. The efferent division, effero, to bring out, of the PNS carries motor commands from the CNS to muscles and glands. These target organs and tissues respond by doing something and are called effectors. Figure 8-1A functional overview of the nervous system. The functions of the central nervous system CNS and peripheral nervous system PNS are shown. Figure 8-1 full alternative text. The efferent division of the PNS has 2 parts. The somatic nervous system, SNS, controls skeletal muscle contractions. Voluntary contractions are under conscious control, such as when you lift a glass of water to your lips. Involuntary contractions are simple or complex movements controlled at the subconscious level. For example, if you accidentally place your hand on a hot stove, you will pull it back immediately, even before you notice any pain. This type of automatic response is called a reflex. The autonomic nervous system, ANS, or visceral motor system, automatically regulates smooth muscle, cardiac muscle, glandular secretions, and adipose tissue at the subconscious level. The ANS includes a sympathetic division and a parasympathetic division, which commonly have opposite effects. For example, activity of the sympathetic division speeds up your heart rate, but the parasympathetic division slows your heart rate. The ANS also includes a third division, called the enteric nervous system, ENS. The ENS is the network of nervous tissue in the walls of the digestive tract. It helps control digestive functions independent of the CNS. We consider its role in Chapter 16. Checkpoint. Identify the two anatomical divisions of the nervous system. Identify the two functional divisions of the peripheral nervous system, and describe their primary functions. What would be the effect of damage to the afferent division of the PNs? See the Blue Answers tab at the back of the book. 8 to 2 neurons are specialized for intercellular communication and are supported by cells called neuroglia. Learning outcome. Distinguish between neurons and neuroglia on the basis of structure and function. The nervous system includes all the neural tissue in the body. Neural tissue, introduced in Chapter 4, consists of two kinds of cells, neurons and neuroglia. 113 neurons, nerve, are the basic functional units of the nervous system. All neural functions involve the communication of neurons with one another and with other cells. The neuroglia, glia, blue, regulate the environment around neurons, provide a supporting framework for neural tissue, and act as phagocytes. Although they are much smaller cells, neuroglia, also called glial cells, far outnumber neurons. Unlike most neurons, most glial cells retain the ability to divide. Neurons. The general structure of neurons. Neurons can have a variety of shapes. Figure 8-2 shows a multipolar neuron, the most common type of neuron in the CNS. A multipolar neuron has 1. A cell body, 2. Several branching, sensitive dendrites, which receive incoming signals, and 3. A single long axon, which carries outgoing signals toward 4. One or more axon terminals. Figure 8-2 the anatomy of a representative neuron. The relationships between the anatomical structures of a neuron. The relationships of the four parts of a neuron, dendrites, cell body, axon, and axon terminals, are shown in the multipolar neuron depicted here. Figure 8-2 full alternative text. The cell body contains a large, round nucleus with a prominent nucleolus. The cytoplasm of the cell body contains organelles that provide energy and synthesize organic compounds. The numerous mitochondria, free and fixed ribosomes, and membranes of the rough endoplasmic reticulum, rare, give the cytoplasm a coarse, grainy appearance. Clusters of rare and free ribosomes are known as Nislo bodies. They give a gray color to areas containing neuron cell bodies and account for the color of gray matter seen in brain and spinal cord dissections. Projecting from the cell body are a variable number of dendrites and a single large axon. The plasma membrane of the dendrites and cell body is sensitive to chemical, mechanical, or electrical stimulation. In a process described later, such stimulation often leads to the generation of an electrical impulse, or action potential, that travels along the axon. Action potentials begin at a thickened region of the cell body called the axon hillock. The axon may branch along its length, producing branches called collaterals. Axon terminals, also called synaptic terminals and synaptic knobs, are found at the tips of each branch. An axon terminal is part of a synapse, a site where a neuron communicates with another cell. Most neurons lack centrioles, organelles involved in the movement of chromosomes during mitosis. P70 as a result, typical CNS neurons cannot divide, so they cannot be replaced if lost to injury or disease. Neural stem cells are present in the adult nervous system, but they are typically inactive. Exceptions occur in the nose, where the regeneration of olfactory, smell, receptors maintains our sense of smell, and in the hippocampus, a portion of the brain involved in storing memories. Researchers are studying the processes that trigger neural stem cell activity, with the goal of preventing or reversing neuron loss due to trauma, disease, or aging. Structural classification of neurons. The billions of neurons in the nervous system are variable in form. Based on the relationship of the dendrites to the cell body and axon, neurons are classified into 3 types, figure 8-3. Figure 8-3 a structural classification of neurons. The structure of multipolar, unipolar and bipolar neurons. The neurons are not drawn to scale. Bipolar neurons are many times smaller than typical unipolar and multipolar neurons. The arrows show the normal direction of an action potential. Figure 8-3 full alternative text. A multipolar neuron has 2 or more dendrites and a single axon, figure 8-3a. These are the most common neurons in the CNS. All the motor neurons that control skeletal muscles are multipolar. Their axons may be a meter or more in length. In a unipolar neuron, the dendrites and axon are continuous, and the cell body lies off to one side, figure 8-3b. In a unipolar neuron, the action potential begins at the base of the dendrites, and the rest of the process is considered an axon. Most sensory neurons of the peripheral nervous system are unipolar. Their axons can be as long as those of multipolar neurons. Bipolar neurons have only one dendrite and one axon, with the cell body between them, figure 8-3c. Bipolar neurons are small and rare. They occur in special sense organs, where they relay information about sight, smell, or hearing from receptor cells to other neurons. Functional classification of neurons. In terms of function, neurons are sorted into 3 groups: 1. Sensory neurons, 2. Motor neurons, and 3. Interneurons. Sensory neurons. The approximately 10 billion sensory neurons, or afferent neurons, in the human body form the afferent division of the PNs. Sensory neurons receive information from sensory receptors monitoring the external and internal environments and then relay the information to other neurons in the CNS, spinal cord or brain. The receptor may be a dendrite of a sensory neuron or specialized cells of other tissues that communicate with the sensory neuron. Receptors may be categorized according to the information they detect. 2 types of somatic sensory receptors detect information about the outside world or our physical position within it. External receptors provide information about the external environment in the form of sensations of touch, pressure, pain, and temperature, and the more complex senses of taste, smell, sight, equilibrium, and hearing. Proprioceptors, proprios, one's own plus capio, to take, monitor the position and movement of skeletal muscles and joints. Visceral receptors, or internal receptors, monitor the activities of the digestive, respiratory, cardiovascular, urinary, and reproductive systems. They provide sensations of distension, deep pressure, and pain. Motor neurons. The half-million motor neurons, or efferent neurons, of the efferent division carry instructions from the CNS to other tissues, organs, or organ systems. These peripheral targets are called effectors. For example, a skeletal muscle is an effector that contracts when it receives neural stimulation. Neurons in the two efferent divisions of the PNs target separate classes of effectors. The somatic motor neurons of the somatic nervous system innervate skeletal muscles, and the visceral motor neurons of the autonomic nervous system innervate all other effectors, including cardiac muscle, smooth muscle, and glands. Interneurons. The 20 billion interneurons, or association neurons, are located entirely within the brain and the spinal cord. Interneurons, as the name implies, between, interconnect other neurons. They are responsible for distributing sensory information and coordinating motor activity. The more complex the response to a given stimulus, the greater the number of interneurons involved. Interneurons also play a role in all higher functions, such as memory, planning, and learning. Neuroglia are abundant and diverse. They make up about half of the volume of the nervous system. They are found in both the CNS and PNs, but the CNS has a greater variety of glial cells. The CNS contains 4 types of neuroglial cells, figure 8-4. Figure 8-4 neuroglia in the CNS. The relationship between neuroglia and neurons in the nervous tissue and their surrounding structures are identified. This diagrammatic view of nervous tissue in the CNS depicts the relationships between neuroglia and neurons. Figure 8-4 full alternative text. Astrocytes, astrocytes, star plus cyte, cell, are the largest and most numerous neuroglia. These star-shaped cells have varied functions. Astrocytes maintain the blood-brain barrier that isolates the CNS from the body's general circulation. Cytoplasmic extensions of the astrocytes end in expanded feet that wrap around capillaries. The astrocytes secrete chemicals that cause the capillaries of the CNS to become impermeable to many compounds, such as hormones and amino acids that could interfere with neuron function. Astrocytes also create a structural framework for CNS neurons and perform repairs in damaged neural tissues. Oligodendrocytes, oligodendrocytes, oligo-few, have smaller cell bodies and fewer processes, cytoplasmic extensions, than astrocytes. The plasma membrane at the tip of each process forms a thin, expanded sheet that wraps around an axon. This membranous wrapping is called myelin. It serves as electrical insulation that increases the speed at which an action potential travels along the axon. Each oligodendrocyte myelinates short segments of several axons, so many oligodendrocytes are needed to coat an entire axon with myelin. Such an axon is said to be myelinated. The areas covered in myelin are called internodes. The small gaps between adjacent cell processes are called nodes, or the nodes of Ranvier. Myelin is lipid-rich, and on dissection, areas of the CNS containing myelinated axons appear glossy white. Areas dominated by myelinated axons are known as the white matter of the CNS. Not every axon in the CNS is myelinated. Axons without a myelin coating are said to be unmyelinated. Areas containing neuron cell bodies, dendrites, and unmyelinated axons make up the gray matter of the CNS. Microglia are the smallest and least numerous of the neuroglia in the CNS. Microglia are phagocytic cells derived from white blood cells that migrated into the CNS as the nervous system formed. They perform protective functions such as engulfing cellular waste and pathogens. Ependymal cells are simple cuboidal epithelial cells that line cavities in the CNS filled with cerebrospinal fluid, CSF. These cavities include the central canal of the spinal cord and the chambers, or ventricles, of the brain. The lining of epithelial cells is called the ependyma, ependyma. Unlike other epithelia, it lacks a basement membrane. In some regions of the brain, the ependyma produces CSF. In other locations, the cilia on ependymal cells help circulate this fluid within and around the CNS. The PNs contains 2 types of neuroglia. Satellite cells surround and support neuron cell bodies in the peripheral nervous system, much as astrocytes do in the CNS. The other glial cells in the PNs are Schwann cells, also called neurolomocytes, figure 8-5a. Figure 8-5 Schwann cells and peripheral axons. The structures of Schwann cells on peripheral axons in the PNS. Figure 8-5 full alternative text. Schwann cells cover every axon outside the CNS. Wherever a Schwann cell covers an axon, the outer surface of the Schwann cell is called the neurolemma. Unlike an oligodendrocyte in the CNS, which may myelinate portions of several axons, a Schwann cell can myelinate only one segment of a single axon, figure 8-5a. However, a Schwann cell can enclose portions of several different unmyelinated axons, figure 8-5b. Organization of neurons in the nervous system. Neuron cell bodies and their axons are not randomly scattered in the CNS and PNs. Instead, they are organized into masses or bundles that have distinct anatomical boundaries and are identified by specific terms, figure 8-6. We will use these terms again, so you may find a brief overview here helpful. Figure 8-6 anatomical organization of the nervous system. The organization of the PNS, CNS and pathways of effectors and receptors. Figure 8-6 full alternative text. In the PNs. Neuron cell bodies, gray matter, are located in ganglia, singular, ganglion. The neuron cell bodies are surrounded by satellite cells. The white matter of the PNs contains axons bundled together in nerves. Spinal nerves are connected to the spinal cord, and cranial nerves are connected to the brain. Both sensory and motor axons may be present in the same nerve. In the CNS, A collection of neuron cell bodies, gray matter, with a common function is called a center. A center with a discrete boundary is called a nucleus. Portions of the brain surface are covered by a thick layer of gray matter called neural cortex, rind. The term higher centers refers to the most complex integration centers, nuclei, and areas of cortex in the brain. The white matter of the CNS contains bundles of axons that share common origins, destinations, and functions. These bundles are called tracts. Tracts in the spinal cord form larger groups called columns. Pathways include both gray matter and white matter. They link the centers of the brain with the rest of the body. For example, sensory, ascending, pathways distribute information from sensory receptors to processing centers in the brain. Motor, descending, pathways begin at CNS centers for motor activity and end at the skeletal muscles they control. Clinical note: demyelination disorders. Demyelination is the progressive destruction of myelin sheaths, both in the CNS and PNs. The result is a gradual loss of sensation and motor control that leaves affected body regions numb and paralyzed. One demyelination disorder is multiple sclerosis, hardness, or MS. It affects axons in the optic nerve, brain, and/or spinal cord. Common signs and symptoms of MS include partial loss of vision and problems with speech, balance, and general motor coordination. Other important demyelination disorders include heavy metal poisoning, Charcot-Marie-Tooth disease, and Guillain-Barré syndrome. Checkpoint. Name the structural components of a typical neuron. Examination of a tissue sample reveals unipolar neurons. Are these more likely to be sensory neurons or motor neurons? Identify the neuroglia of the central nervous system. Which type of glial cell would increase in number in the brain tissue of a person with a CNS infection? In the PNs, neuron cell bodies are located in and surrounded by neuroglial cells called cells. See the Blue Answers tab at the back of the book. 8 to 3 in neurons, a change in the plasma membrane's electrical potential may result in an action potential, nerve impulse. Learning outcome. Describe the events involved in the generation and propagation of an action potential. The sensory, integrative, and motor functions of the nervous system are dynamic and ever-changing. All communications between neurons and other cells take place through their membrane surfaces. These membrane changes are electrical events that proceed at great speed. The membrane potential. A characteristic feature of all living cells is a polarized plasma membrane. An undisturbed, or unstimulated, cell has a plasma membrane that is polarized because the membrane separates an excess of positive charges outside the cell from an excess of negative charges inside the cell. When positive and negative charges are held apart, a potential difference is said to exist between them. This potential difference is called a membrane potential, or transmembrane potential, because the charges are separated by a plasma membrane. The unit of measurement of potential difference is the volt, V. Most cars, for example, have 12V batteries. The membrane potential of cells is much smaller and is usually reported in millivolts, MV, thousandths of a volt. The membrane potential of an unstimulated, resting cell is known as its resting membrane potential. The resting membrane potential of a neuron is 70 MV. The minus sign indicates that the cytoplasmic surface of the plasma membrane contains an excess of negative charges compared to the extracellular surface. Factors responsible for the membrane potential. Many factors influence membrane potential. In addition to an imbalance of electrical charges, the intracellular and extracellular fluids differ markedly in chemical and ionic composition. For example, the extracellular fluid contains relatively high concentrations of sodium ions, Na+, and chloride ions, Cl. The intracellular fluid contains high concentrations of potassium ions, K+, and negatively charged proteins, Pr. The selective permeability of the plasma membrane maintains these differences between the intracellular and extracellular fluids. The proteins within the cytoplasm are too large to cross the membrane. The ions can enter or leave the cell only with the aid of membrane channels and/or carrier proteins. P61. There are many different types of membrane channels. Some, called leak channels, are always open. Others, called gated channels, open or close under specific circumstances. For example, gated channels may open or close due to the presence of a specific chemical, ion, or a change in membrane potential, or voltage. Both passive and active processes act across the plasma membrane to determine the membrane potential at any moment. The passive forces are chemical and electrical. Chemical concentration gradients move potassium ions out of the cell and sodium ions into the cell. These ions move through separate leak channels. However, it is easier for potassium ions to diffuse through a potassium channel than for sodium ions to diffuse through sodium channels. As a result, potassium ions diffuse out of the cell faster than sodium ions enter the cell. Electrical forces across the membrane also affect the passive movement of sodium and potassium ions. The overall positive charge on the outer surface of the plasma membrane repels positively charged potassium ions. At the same time, the negatively charged inner membrane surface attracts the positively charged sodium ions. Potassium ions continue to leave the cell, however, because its chemical concentration gradient is stronger than the repelling electrical force. To maintain a potential difference across the plasma membrane, active processes work both to overcome the combined chemical and electrical forces driving sodium ions into the cell and to maintain the potassium concentration gradient. The resting potential remains stable over time because of the actions of a carrier protein, the sodium-potassium exchange pump. P. 66. This ion pump exchanges 3 intracellular sodium ions for 2 extracellular potassium ions. At the normal resting membrane potential of 70 MV, sodium ions are ejected as fast as they enter the cell. The cell, therefore, undergoes a net loss of positive charges. As a result, the interior surface of the plasma membrane maintains an excess of negative charges, primarily from negatively charged proteins. Figure 8-7 shows the plasma membrane at the resting membrane potential. Figure 8-7 the resting membrane potential. The arrangement of ions in a resting membrane potential. Figure 8-7 full alternative text. Changes in the membrane potential. Any stimulus that 1. Alters membrane permeability to sodium or potassium ions, or 2. Alters the activity of the exchange pump will disturb the resting membrane potential of a cell. Some stimuli that can affect membrane potential include exposure to specific chemicals, mechanical pressure, changes in temperature, or shifts in the extracellular ion concentrations. Any change in the resting potential can have an immediate effect on the cell. For example, permeability changes in the sarcolemma of a skeletal muscle fiber trigger a contraction. p. 199. In most cases, a stimulus opens gated ion channels that are closed when the plasma membrane is at its resting membrane potential. The opening of these channels speeds up ion movement across the plasma membrane and changes the membrane potential. For example, the opening of gated sodium ion channels speeds up the entry of sodium ions, Na+, into the cell. As the number of positively charged ions on the inner surface of the plasma membrane increases, the membrane potential shifts towards 0 MV. A shift in this direction is called a depolarization of the membrane. On the other hand, a stimulus that opens gated potassium ion channels shifts the membrane potential away from 0 MV, because additional potassium ions, K+, will leave the cell. Such a change may take the membrane potential from 70 MV to 80 MV. This kind of shift is called a hyperpolarization. Information transfer between neurons and other cells involves two types of change in membrane potential, graded potentials and action potentials. Graded potentials, or local potentials, are changes in the membrane potential that cannot spread far from the site of stimulation. For example, if a chemical stimulus to the plasma membrane of a neuron opens gated sodium ion channels at a single site, the Na+ entering the cell will depolarize the membrane at that location. Attracted to surrounding negative ions, the Na+ move along the inner surface of the membrane in all directions. The degree of depolarization decreases with distance from the stimulation site. What? This happens because the cytosol resists ion movement and because some sodium ions are lost as they move back out across the membrane through leak channels. Graded potentials occur in the plasma membranes of all cells in response to environmental stimuli. They often trigger specific cell functions. For example, a graded potential in the membrane of a gland cell may trigger secretion. However, graded potentials affect too small an area to have an effect on the activities of such large cells as skeletal muscle fibers or neurons. In these cells, graded potentials can influence activities in distant portions of the cell only if they generate an action potential, an electrical signal that affects the surface of the entire membrane. An action potential is a propagated change in the membrane potential of excitable cells. Only excitable cells have an electrically excitable membrane that can be stimulated to propagate action potentials. Excitable membranes contain voltage-gated ion channels that open or close in response to changes in the membrane potential. Skeletal muscle fibers, cardiac muscle cells, and the axons of neurons have excitable membranes. In a skeletal muscle fiber, the action potential begins at the neuromuscular junction and travels along the entire membrane surface, including the T-tubules. P204 the resulting ion movements trigger a contraction. In an axon, an action potential usually begins near the axon hillock and travels along the length of the axon toward the axon terminals, where its arrival activates the synapses. An action potential in a neuron is also known as a nerve impulse. We discuss action potentials in cardiac muscle cells in Chapter 12. Action potentials are generated by the opening and closing of voltage-gated Na+ channels and voltage-gated K+ channels in response to a graded potential. This local depolarization acts like pressure on the trigger of a gun. The gun fires only after a certain minimum pressure has been applied to the trigger. It does not matter whether the pressure builds gradually or is exerted suddenly. When the pressure reaches a critical point, the gun will fire. Whenever the gun fires, the forces that were applied to the trigger have no effect on the speed of the bullet leaving the gun. In an axon, the graded potential is the pressure on the trigger, and the action potential is like the firing of the gun. An action potential will not appear unless the membrane depolarizes to a level known as the threshold. Every stimulus, whether minor or extreme, that brings the membrane to threshold will generate an identical action potential. This concept is called the all or none principle because a given stimulus either triggers a typical action potential or none at all. The all or none principle applies to all excitable membranes. Build your knowledge. Recall that a single axon of a motor neuron may branch to control more than one skeletal muscle fiber, as you saw in Chapter 7, the muscular system. Each branch ends in an axon terminal that is part of a neuromuscular junction. Each skeletal muscle fiber has only one neuromuscular junction. A motor unit is a single motor neuron and all the muscle fibers it innervates. P. 205. The generation of an action potential. How is an action potential generated? An action potential begins when the first portion of the excitable axon membrane, called the initial segment, depolarizes to threshold. The steps involved in generating an action potential begin with a graded depolarization to threshold, from 70 MV to 60 MV, and end with a return to the resting membrane potential, 70 MV. The steps are illustrated in Spotlight Figure 8-8. Spotlight Figure 8-8 the generation of an action potential. The steps involved in generating an action potential. A neuron receives information on its dendrites and cell body, and communicates that information to another cell at its axon terminal. Because the two ends of the neuron may be a meter apart, such long-range communication relies on action potentials. Action potentials are propagated changes in the membrane potential that, once started, affect the entire excitable membrane of the axon. Action potentials depend on the presence of voltage-gated sodium and potassium ion channels. Steps in the generation of an action potential in an axon. The first step is a graded depolarization caused by the opening of chemically gated sodium ion channels, usually at the axon hillock. The axon membrane colors in steps 1 to 4 match the colors of the line graph showing changes in the membrane potential. Go to Mastering ANP Study Area Menu MP3 Tutor Session Generation of an Action Potential. Figure 8-8 Full Alternative Text. The membrane cannot respond normally to further stimulation during most of these steps. This period is known as the refractory period of the membrane. It lasts from the moment the voltage-gated sodium channels open at threshold until the return to the resting potential, or repolarization, is complete. The refractory period limits the rate at which action potentials can be generated in an excitable membrane. The maximum rate of action potential generation is 500 to 1000 per second. Propagation of an action potential. An action potential initially involves a relatively small portion of the total membrane surface of the axon. But unlike braided potentials, which diminish rapidly with distance, action potentials affect the entire membrane surface. The basic processes of action potential propagation along unmyelinated and myelinated axons are shown in Spotlight Figure 8-9, P256. For simplicity, think of the plasma membrane as a series of adjacent segments. Spotlight Figure 8-9A. Spotlight Figure 8-9 Propagation of an action potential. The basic processes of action potential propagation along unmyelinated and myelinated axons. Figure 8-9 Full alternative text. The action potential begins at the axon's initial segment. For a brief moment at the peak of the action potential, the membrane potential becomes positive rather than negative. A local current then develops as the sodium ions begin moving in the cytosol and the extracellular fluid. The local current of moving sodium ions spreads in all directions, depolarizing adjacent portions of the membrane. The axon hillock cannot respond with an action potential because it does not have voltage-gated sodium ion channels. The process then continues in a chain reaction, and each time a local current develops, the action potential moves forward along the axon. It does not move backward, because the previous segment of the axon is still in the refractory period. As a result, an action potential always proceeds away from its generation site and cannot reverse direction. Eventually, it reaches the most distant portions of the plasma membrane. This form of action potential transmission is known as continuous propagation. You might compare continuous propagation to a person walking towards some destination by taking small baby steps. Progress is made, but slowly. Continuous propagation takes place along unmyelinated axons at a speed of about 1 meter per second, 2 miles per hour. In a myelinated fiber, the axon is wrapped in myelin. This wrapping is complete except at the nodes, where adjacent glial cells contact one another. Between the nodes, the lipids of the myelin sheath block the flow of ions across the membrane. As a result, continuous propagation cannot take place. Instead, when an action potential is initiated by the axon hillock, the local current skips the internode and depolarizes the closest node to threshold. Spotlight Figure 8-9b. In this way, the action potential jumps from node to node rather than proceeding in a series of small steps. This jumping process is called saltatory propagation. Its name comes from salter, the Latin word meaning to leap. Saltatory propagation carries nerve impulses along an axon at speeds ranging from 18 to 140 meters per second, 40 to 300 miles per hour. This faster process might be compared to a person jumping over puddles on the way to a destination. Checkpoint. What effect would a chemical that blocks the voltage-gated sodium ion channels in the excitable axon membrane of a neuron have on its ability to depolarize? What effect would decreasing the concentration of extracellular potassium ions have on the membrane potential of a neuron? List the steps involved in the generation of an action potential. Two axons are tested for propagation velocities, speeds. One carries action potentials at 50 meters per second, the other at 1 meter per second. Which axon is myelinated? See the Blue Answers tab at the back of the book. 8-4 at synapses, communication takes place among neurons or between neurons and other cells. Learning outcome. Describe the structure of a synapse, and explain the process of nerve impulse transmission at a synapse. Recall that a synapse is a site where a neuron communicates with another cell. In the nervous system, information moves from one location to another in the form of action potentials, nerve impulses, along axons. At the end of an axon, the arrival of an action potential results in the transfer of information to another neuron or to an effector cell. The information transfer takes place through the release of chemicals called neurotransmitters from the axon terminal. The synapse where one neuron communicates with another may be on a dendrite, on the cell body, or along the length of the axon. Synapses between a neuron and another cell type are called neuroeffector junctions. As you have learned, a neuron communicates with a muscle cell at a neuromuscular junction. P197 at a neuroglandular junction, a neuron controls or regulates the activity of a secretory cell. Structure of the synapse. Communication between neurons and other cells takes place in only one direction across a synapse. At a synapse between two neurons, the nerve impulse passes from the axon terminal of the sending neuron, called the presynaptic neuron, to the receiving neuron, called the postsynaptic neuron, figure 8-10. The opposing plasma membranes are separated by a narrow space called the synaptic cleft. Figure 8-10 The structure of a typical synapse. The structures in a typical synapse between two neurons. A diagrammatic view of a typical synapse between two neurons. Figure 8-10 Full alternative text. Each axon terminal contains synaptic vesicles. Each vesicle contains several thousand molecules of a specific neurotransmitter. Upon stimulation, many of these vesicles release their contents into the synaptic cleft. The neurotransmitter then diffuses across the synaptic cleft and binds to receptors on the postsynaptic membrane. Synaptic function and neurotransmitters. There are many different neurotransmitters. The neurotransmitter acetylcholine, or ACH, is released at cholinergic synapses. These synapses are widespread inside and outside of the CNS. The neuromuscular junction, described in Chapter 7, is one example. Figure 8-11 shows the major events that take place at a cholinergic synapse after an action potential arrives at the presynaptic neuron. Figure 8-11 The events at a cholinergic synapse. The major events that take place at a cholinergic synapse after an action potential arrives at the presynaptic neuron. Figure 8-11 Full alternative text. An action potential arrives and depolarizes the axon terminal. The axon terminal releases the neurotransmitter ACH. Depolarization of the presynaptic membrane causes voltage-gated calcium ion channels to open briefly, allowing extracellular calcium ions to enter the axon terminal. Their entry triggers the exocytosis of the synaptic vesicles and the release of ACH. ACH release stops very quickly, because active transport processes rapidly remove the calcium ions from the cytosol. ACH binds to ACH receptors and depolarizes the postsynaptic membrane. ACH binding causes these chemically gated Na+ channels to open and allow sodium ions to enter. If the resulting depolarization of the postsynaptic membrane reaches threshold, an action potential is generated. ACH is removed by ACE. The effects on the postsynaptic membrane are temporary. The reason is because the synaptic cleft and postsynaptic membrane contain the enzyme acetylcholinesterase, ACE. P197 removes ACH by breaking it into acetate and choline. Another common neurotransmitter is norepinephrine, norepinephrine, or NE. It is important in the brain and in portions of the autonomic nervous system. NE is also called noradrenaline, and synapses releasing NE are described as adrenergic. The neurotransmitters dopamine, gamma-aminobutyric, gamma-aminobutyric acid, also known as GABA, and serotonin, function in the CNS. There are at least 50 other neurotransmitters whose functions are not well understood. In addition, two gases are important neurotransmitters, nitric oxide, NO, and carbon monoxide, CO. The neurotransmitters released at a synapse may have excitatory or inhibitory effects. Both ACH and NE usually have an excitatory, depolarizing effect on postsynaptic neurons. Like ACH, NE has a temporary effect. It is broken down by an enzyme called monoamine oxidase. The effects of dopamine, GABA, and serotonin are usually inhibitory because they tend to hyperpolarize postsynaptic neurons. Many drugs affect the nervous system by stimulating receptors that otherwise respond only to neurotransmitters. These drugs can have complex effects on perception, motor control, and emotional states. Whether or not an action potential appears in the postsynaptic neuron depends on the balance between the depolarizing and hyperpolarizing stimuli arriving at any moment. For example, suppose that a neuron will generate an action potential if it receives 10 depolarizing stimuli. That could mean 10 active synapses if all release excitatory neurotransmitters. But if, at the same moment, 10 other synapses release inhibitory neurotransmitters, the excitatory and inhibitory effects will cancel one another out. As a result, no action potential would develop. The activity of a neuron thus depends on the balance between excitation and inhibition. The interactions are extremely complex. Synapses at the cell body and dendrites may involve tens of thousands of other neurons. Some release excitatory neurotransmitters and others release inhibitory neurotransmitters. Neuronal pools. As noted earlier, the human body has about 10 million sensory neurons, 20 billion interneurons, and 1/2 million motor neurons. These individual neurons represent the simplest level of organization within the CNS. However, the integration of sensory and motor information to produce complex responses requires groups of interneurons acting together. A neuronal pool is a group of interconnected interneurons with specific functions. Each neuronal pool has a limited number of input sources and output destinations. Also, each pool may contain excitatory and inhibitory neurons. The output of one pool may stimulate or depress the activity of other pools, or it may exert direct control over motor neurons or peripheral effectors. Neurons and neuronal pools communicate with one another in several wiring diagrams, or neural circuits. The two simplest circuit patterns are divergence and convergence. In divergence, information spreads from one neuron to several neurons, or from one neuronal pool to multiple neuronal pools, figure 8-12a. Considerable divergence occurs when sensory neurons bring information into the CNS. The sensory information is distributed to different neuronal pools throughout the spinal cord and brain. For example, visual information from the eyes reaches your conscious awareness at the same time it is carried to areas of the brain that control posture and balance at the subconscious level. Figure 8-12 Two common types of neuronal pools. The two common types of neuronal pools are convergence and divergence. Figure 8-12 Full alternative text. Divergence is also involved when you step on a sharp object. That action stimulates sensory neurons that distribute information to a number of neuronal pools. As a result, you might withdraw your foot, shift your weight, move your arms, feel the pain, and shout out, all at the same time. In convergence, figure 8-12b, several neurons synapse on a single postsynaptic neuron. Convergence makes possible both voluntary and involuntary control of some body processes. For example, as you read this, the movements of your diaphragm and ribs are being involuntarily, or subconsciously, controlled by respiratory centers in your brain. These centers activate or inhibit motor neurons in your spinal cord that control your respiratory muscles. But those same movements can be controlled voluntarily, or consciously, as when you take a deep breath and hold it. Two neuronal pools are involved, and both synapse on the same motor neurons. Checkpoint. Describe the general structure of the synapse. What effect would blocking calcium ion channels at a cholinergic synapse have on synapse function? What type of neural circuit permits both conscious and subconscious control of the same motor neurons? See the Blue Answers tab at the back of the book. 8-5 The brain and spinal cord are surrounded by 3 layers of membranes called the meninges. Learning outcome. Describe the 3 meningeal layers that surround the central nervous system. The central nervous system, CNS, consists of the spinal cord and brain. The nervous tissue of the CNS is delicate. It also has a very high metabolic rate and requires abundant nutrients and a constant supply of oxygen. At the same time, CNS tissue must be isolated from a variety of compounds in the blood that could interfere with its complex operations. It also needs to be protected against damaging contact with the surrounding bones. The meninges, meninges, are 3 layers of specialized membranes surrounding the brain and spinal cord, figure 8-13. They provide the CNS tissue with physical stability and shock absorption. Blood vessels branching within these layers deliver needed oxygen and nutrients. At the foramen magnum of the skull, the cranial meninges covering the brain are continuous with the spinal meninges that surround the spinal cord. The meninges cover cranial nerves as they extend through foramina of the skull and spinal nerves that pass through the intervertebral foramina. The meninges then become continuous with the connective tissue surrounding the peripheral nerves. Figure 8-13 The meninges of the brain and spinal cord. The location and structure of the cranial and spinal meninges. Figure 8-13 Full alternative text. The 3 meningeal layers are the dura mater, the arachnoid, and the pia mater. The dura mater. The tough, fibrous dura mater, hard plus mater, mother, forms the outermost covering of the central nervous system. The dura mater surrounding the brain consists of two fibrous layers. The outer layer is fused to the periosteum of the skull. In some places, the inner layer is joined with the outer layer. Typically, the two layers are separated by a slender gap that contains tissue fluids and blood vessels, figure 8-13a. At several locations, the inner layer of the dura mater extends deep into the cranial cavity, forming folded membranous sheets called dural folds. The dural folds act like seat belts to hold the brain in position. Large collecting veins known as dural sinuses lie between the two layers of a dural fold. In the spinal cord, the outer layer of the dura mater is not fused to bone. An epidural space lies between the dura mater of the spinal cord and the walls of the vertebral canal. This space contains areolar tissue, blood vessels, and adipose tissue, figure 8-13b. Injecting an anesthetic into the epidural space produces a temporary sensory and motor paralysis known as an epidural block. This technique has the advantage of affecting only the spinal nerves in the immediate area of the injection. Epidural blocks in the lower lumbar or sacral regions may be used to control pain during childbirth. The arachnoid. A narrow subdural space separates the inner surface of the dura mater from the second meningeal layer, the arachnoid, arachne, spider. This intervening space contains a small quantity of lymphatic fluid, which reduces friction between the opposing surfaces. The arachnoid is a layer of squamous epithelial cells. Deep to this layer lies the subarachnoid space, which contains a delicate web of collagen and elastic fibers. The subarachnoid space is filled with cerebrospinal fluid, CSF. This fluid acts as a shock absorber and transports dissolved gases, nutrients, chemical messengers, and waste products. The pia mater. The subarachnoid space separates the arachnoid from the innermost meningeal layer, the pia mater, pia, delicate plus mater, mother. The pia mater is bound firmly to the underlying neural tissue. The blood vessels servicing the brain and spinal cord run along the surface of this layer, within the subarachnoid space. The pia mater of the brain is highly vascular, and large blood vessels branch over the surface of the brain, supplying the superficial areas of neural cortex. This extensive blood supply is extremely important, for the brain has a very high rate of metabolism. At rest, the 1.4 kilograms 3.1 pounds brain uses as much oxygen as 28 kilograms 61.6 pounds of skeletal muscle. Clinical note: epidural and subdural hemorrhages. A severe head injury may damage cerebral blood vessels and cause bleeding into the cranial cavity. If blood is forced between the dura mater and the skull, the condition is known as an epidural hemorrhage. A subdural hemorrhage is bleeding between the dura mater and the arachnoid mater. These conditions are serious because the blood entering the epidural or subdural spaces compresses and distorts the relatively soft tissues of the brain. The signs and symptoms vary depending on whether an artery or a vein is damaged. Because arterial blood pressure is higher, bleeding from an artery can cause more rapid and severe distortion of neural tissue. There is good reason for adults and children to give their heads added protection by wearing helmets while biking, motorcycling, skiing, skateboarding, and playing various sports. A bicycle helmet. Checkpoint. Identify the three meninges surrounding the CNS. See the Blue Answers tab at the back of the book. 8-6 The spinal cord contains gray matter surrounded by white matter and connects to 31 pairs of spinal nerves. Learning outcome. Discuss the roles of gray matter and white matter in the spinal cord. The spinal cord serves as the major highway for sensory impulses passing to the brain and motor impulses coming from the brain. In addition, the spinal cord integrates information on its own. It controls spinal reflexes, automatic motor responses ranging from withdrawal from pain to complex reflex patterns involved in sitting, standing, walking, and running. Gross anatomy. The adult spinal cord, figure 8-14a, is approximately 45 centimeters 18 inches long and has a maximum width of roughly 14 millimeters 0.55 inches. With two exceptions, the diameter of the spinal cord decreases as it extends inferiorly. The two exceptions are regions concerned with the sensory and motor control of the limbs. The cervical enlargement supplies nerves to the shoulder girdles and upper limbs. The lumbosacral enlargement provides innervation to the pelvis and lower limbs. Inferior to the lumbosacral enlargement, the spinal cord becomes tapered and conical. A slender strand of fibrous tissue extends from the inferior tip of the spinal cord to the coccyx, serving as an anchor that prevents upward movement. Figure 8-14 Gross anatomy of the spinal cord. The cervical, thoracic, lumbar, and sacral regions of the spinal cord are labeled. Figure 8-14 Full alternative text. The spinal cord has a central canal, a narrow internal passageway filled with cerebrospinal fluid, figure 8-14b. The posterior surface of the spinal cord has a shallow groove, the posterior median sulcus. The anterior surface has a deeper groove, the anterior median fissure. The entire spinal cord consists of 31 segments. Each gives rise to a pair of spinal nerves. There are 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and 1 coccygeal spinal segments, figure 8-14c. Each pair of spinal nerves is identified by a letter and number, figure 8-14ac. Every spinal segment is associated with a pair of spinal ganglia, singular, ganglion, which contain the cell bodies of sensory neurons, figure 8-14b. The posterior roots contain the axons of these neurons and bring sensory information to the spinal cord. A pair of anterior roots contains the axons of CNS motor neurons that control muscles and glands. On either side, the posterior and anterior roots from each segment leave the vertebral column between adjacent vertebrae at the intervertebral foramen. Distal to each spinal ganglion, the sensory, posterior, and motor, anterior, roots are bound together into a single spinal nerve. All spinal nerves are classified as mixed nerves because they contain both sensory and motor fibers. The spinal nerves on either side form outside the vertebral canal, where the anterior and posterior roots unite. P. 162. The spinal cord continues to grow until a person is approximately 4 years old. After age 4, the vertebral column continues to elongate, but the spinal cord does not. As a result, the posterior and anterior roots gradually grow longer and the correspondence between the spinal cord segment and its adjacent vertebrae is lost. This explains why spinal cord segments S1 S5 are level with vertebrae T12 L1, figure 8-14c. The adult spinal cord extends only to the level of the first or second lumbar vertebrae. When seen in gross dissection, the long ventral and dorsal roots of spinal nerves inferior to the tip of the spinal cord, plus the cord's thread-like extensions, reminded early anatomists of a horse's tail. With that in mind, they called this complex the cauda equina, cauda ekwna, cauda, tail plus equus, horse. Sectional anatomy. Clinical note: spinal cord injuries. Injuries affecting the spinal cord or cauda equina produce symptoms of sensory loss or motor paralysis that reflect the specific nuclei, tracts, or spinal nerves involved. The general paralysis can result from severe damage to the spinal cord in an auto crash or other accident, and the damaged tracts seldom undergo even partial repairs. Extensive damage at the fourth or fifth cervical vertebra will eliminate sensation and motor control of the upper and lower limbs. The extensive paralysis produced is called quadriplegia. Paraplegia, the loss of motor control of the lower limbs, may follow damage to the thoracic vertebrae. According to the CDC, complete or partial paralysis due to spinal injury affects approximately 200,000 people in the United States. The regeneration of spinal cord tissue was long thought to be impossible because mature nervous tissue had not been observed to grow or undergo mitosis. Biological cures by various means are being sought. One involves interfering with inhibitory factors in the spinal cord that slow the repair of neurons. Another involves implanting or stimulating unspecialized stem cells to grow and divide. Inactive neural stem cells have been found in mature human nervous tissues. This discovery is potentially significant because laboratory rats treated with embryonic stem cells at the site of a spinal cord injury have regained limb mobility and strength. Work also continues on electronic methods of restoring some degree of motor control. In 2014, the FDA approved an electric-powered exoskeleton suit that may help paraplegic individuals walk again. The device consists of a fitted brace that supports the legs and part of the upper body, motors that provide movement at the hips, knees, and ankles, a tilt sensor, and a backpack containing a computer and power supply. Crutches are still needed for additional support. With assistance from a trainer, individuals wearing the exoskeleton will be able to sit, stand, and walk. An adolescent wears forearm crutches as he walks up a set of steps. The anterior median fissure and the posterior median sulcus mark the division between left and right sides of the spinal cord, figure 8 to 15. The gray matter is dominated by the cell bodies of neurons and glial cells. It forms a rough H, or a butterfly shape, around the narrow central canal. Projections of gray matter, called horns, extend outward into the white matter, which contains large numbers of myelinated and unmyelinated axons. Figure 8 to 15 sectional anatomy of the spinal cord. A diagrammatic and micrograph section of the anatomy of the spinal cord is labeled. Figure 8 to 15 full alternative text. Figure 8-15 shows the relationship between the function of a particular nucleus, a collection of sensory or motor cell bodies, and its relative position in the gray matter of the spinal cord. The posterior gray horns contain sensory nuclei. The anterior gray horns are involved in the motor control of skeletal muscles. Nuclei in the lateral gray horns contain the visceral motor neurons that control smooth muscle, cardiac muscle, and glands. The gray chromosomes anterior and posterior to the central canal interconnect the horns on either side of the spinal cord. The gray and white chromosomes contain axons that cross from one side of the spinal cord to the other. The white matter on each side can be divided into 3 regions, or columns, figure 8-15a. The posterior white columns extend between the posterior gray horns and the posterior median sulcus. The anterior white columns lie between the anterior gray horns and the anterior median fissure. The anterior white columns are interconnected by the anterior white chromosomer. The white matter between the anterior and posterior columns makes up the lateral white columns. Each column contains tracts whose axons carry either sensory information or motor commands. Small tracts carry sensory or motor signals between segments of the spinal cord, and larger tracts connect the spinal cord with the brain. Ascending tracts carry sensory information toward the brain. Descending tracts convey motor commands into the spinal cord. Checkpoint. Damage to which root of the spinal nerve would interfere with motor function? A person with polio has lost the use of his leg muscles. In which area of his spinal cord could you locate the poliovirus-infected motor neurons? Why are spinal nerves also called mixed nerves? See the Blue Answers tab at the back of the book. 8 to 7 The brain has several principal structures, each with specific functions. Learning outcome. Name the major regions of the brain, and describe the locations and functions of each. The brain is far more complex than the spinal cord. Its responses to stimuli are also more versatile. The brain contains roughly 20 billion neurons organized into hundreds of neuronal pools. Everything we do and everything we are is the result of brain activity. The adult human brain contains almost 97% of the nervous tissue in the body. A typical adult brain weighs 1.4 kilograms 3 pounds and has a volume of 1,200 cubic centimeters, 71.3. Brain size varies considerably among individuals. The brains of males are, on average, about 10% larger than those of females, because of differences in average body size. There is no correlation between brain size and intelligence. Individuals with the smallest brains, 750 cubic centimeters, or largest brains, 2,100 cubic centimeters, are functionally normal. The major regions of the brain. The adult brain has 6 major regions: 1, the cerebrum; 2, the diencephalon; 3, the midbrain; 4, the pons; 5, the medulla oblongata; and 6, the cerebellum. Major landmarks are indicated in figure 8 to 16. Figure 8 to 16 The brain. Major landmarks in the adult brain are labeled. Figure 8 to 16 Full alternative text. The adult brain is dominated in size by the cerebrum, cerebrum or cerebrum. The cerebrum is made up of large, paired left and right cerebral hemispheres, figure 8-16a. Conscious thoughts, sensations, intellectual functions, memory storage, and processing, and complex movements originate in the cerebrum. The hollow diencephalon, DNSEphalon, via dash, through plus encephalos, brain, is connected to the cerebrum, figure 8-16c. Its largest portion is the thalamus, thalamus. The thalamus contains relay and processing centers for sensory information. The hypothalamus, hypo dash, below, is the floor of the diencephalon. It contains centers involved with emotions, autonomic function, and hormone production. A narrow stalk connects the hypothalamus to the pituitary gland. The pituitary gland is the primary link between the nervous and endocrine systems. We will discuss it in Chapter 10. The epithalamus contains another endocrine structure, the pineal gland. The brainstem contains three major regions of the brain, the midbrain, pons, and medulla oblongata, figure 8-16c. The brainstem contains important processing centers and relay stations for information headed to or from the cerebrum or cerebellum. Nuclei in the midbrain, or mesencephalon, mesencephalon, meso dash, middle, process visual and auditory information and generate involuntary motor responses. This region also contains centers that help maintain consciousness. The pons connects the cerebellum to the brainstem. Pons refers to a bridge. In addition to tracts and relay centers, the pons also contains nuclei involved in somatic and visceral motor control. The pons is also connected to the medulla oblongata, the segment of the brain that is attached to the spinal cord. The medulla oblongata relays sensory information to the thalamus and other brainstem centers. It also contains major centers that regulate autonomic function, such as heart rate, blood pressure, respiration, and digestive activities. The large cerebral hemispheres and the smaller hemispheres of the cerebellum, almost completely cover the brainstem, figure 8-16b. The cerebellum adjusts voluntary and involuntary motor activities on the basis of sensory information and stored memories of previous movements. The ventricles of the brain. Recall that the brain and spinal cord contain internal cavities filled with cerebrospinal fluid and lined by ependymal cells. B. 249 The brain has a central passageway that expands to form four chambers called ventricles, ventricles, figure 8-17. Each cerebral hemisphere contains a large lateral ventricle. These two lateral ventricles are not directly connected. Instead, each communicates with the third ventricle of the diencephalon through an opening, the interventricular foramen. Rather than a ventricle, the midbrain has a slender canal known as the cerebral aqueduct. This passageway connects the third ventricle with the fourth ventricle, which extends into the pons and upper portion of the medulla oblongata. Within the medulla oblongata, the fourth ventricle narrows and becomes continuous with the central canal of the spinal cord. Figure 8-17 The ventricles of the brain. The orientation of the ventricles and their channels in the brain. The orientation and extent of the four ventricles and their connecting channels as they would appear if the brain were transparent. Figure 8-17 Full alternative text. Cerebrospinal fluid. Cerebrospinal fluid, or CSF, fills the ventricles and completely surrounds and bathes the exposed surfaces of the CNS. The CSF has several important functions, including cushioning the brain and spinal cord against physical trauma. Supporting the brain. The brain essentially floats in the cerebrospinal fluid. A human brain weighs about 1,400 g, 3.1 pounds, in air but only about 50 g, 1.8 ounces, when supported by cerebrospinal fluid. Transporting nutrients, chemical messengers, and waste products. Except at the choroid plexus, where CSF is produced, the ependymal lining is freely permeable. The CSF is in constant chemical communication with the interstitial fluid that surrounds the neurons and neuroglia of the CNS. Because free exchange takes place between the interstitial fluid and CSF, changes in CNS function may produce changes in the composition of CSF. Samples of CSF can be obtained through a lumbar puncture, or spinal tap. Such samples provide useful clinical information about CNS injury, infection, or disease. A choroid plexus, choroid, a vascular code plus plexus, a network, is a network of permeable capillaries within each ventricle that produces CSF, figure 8-18a. The capillaries of the choroid plexuses are covered by large ependymal cells that secrete CSF at a rate of about 500 ml/day. The total volume of CSF at any given moment is approximately 150 ml. This means that the entire volume of CSF is replaced roughly every 8 hours. Despite this rapid turnover, the composition of CSF is closely regulated. The rate of removal normally keeps pace with the rate of production. If it does not, a variety of clinical problems may appear. Figure 8-18 The formation and circulation of cerebrospinal fluid. The areas of the brain, brainstem and spinal cord where cerebrospinal fluid is formed, its CNS circulatory, and its absorption into the venous circulation. Figure 8-18 Full alternative text. The areas where cerebrospinal fluid is formed, its circulatory pathway around the CNS, and its absorption into the venous circulation are shown in figure 8-18a. CSF formed at the choroid plexus circulates through the different ventricles and fills the central canal of the spinal cord, and CSF enters the subarachnoid space through openings, apertures, in the roof of the fourth ventricle. Cerebrospinal fluid then flows through the subarachnoid space surrounding the brain, spinal cord, and cauda equina. Between the cerebral hemispheres, slender extensions of the arachnoid mater penetrate the inner layer of the dura mater. Clusters of these extensions form arachnoid granulations, which project into the superior sagittal sinus, a large cerebral vein, figure 8-18b. Diffusion across the arachnoid granulations returns excess cerebrospinal fluid to the venous circulation. The cerebrum. The cerebrum is the largest region of the brain. It is the site where conscious thought and intellectual functions originate. Much of the cerebrum is involved in receiving somatic sensory information and then exerting voluntary or involuntary control over somatic motor neurons. In general, we are aware of these events. However, most sensory processing and all visceral motor, autonomic, control occur elsewhere in the brain, usually outside our conscious awareness. The cerebrum includes gray matter and white matter. Gray matter is found in a superficial layer of neural cortex, known as the cerebral cortex, and in deeper basal nuclei. The white matter lies beneath the neural cortex and surrounds the basal nuclei. Structure of the cerebral hemispheres. A blanket of cerebral cortex ranging from 1 to 4.5 millimeters, 0.04 to 0.18 inches, thick covers the paired cerebral hemispheres, figure 8 to 19. This outer surface forms a series of folds, or gyri, GRI, singular, gyrus. The gyri are separated by shallow depressions, called sulci, sulci, or by deeper grooves, called fissures. Gyri increase the surface area of the cerebrum, and thus the number of neurons in the cortex. The total surface area of the cerebral hemispheres is roughly equivalent to 2,200 square centimeters, 2.5 square feet of flat surface. Figure 8 to 19 motor, sensory, and association areas of the cerebral cortex. The surface of the left cerebral hemisphere shows the associated areas of motor and sensory regions. Major anatomical landmarks on the surface of the left cerebral hemisphere are shown. Retractors along the lateral sulcus reveal the insula. Figure 8 to 19 full alternative text. The two cerebral hemispheres are separated by a deep longitudinal fissure, figure 8-16a. Each hemisphere can be divided into well-defined regions, or lobes, named after the overlying bones of the skull, figures 8-16b and 8 to 19. On each hemisphere, the central sulcus, a deep groove, divides the anterior frontal lobe from the more posterior parietal lobe. The horizontal lateral sulcus separates the frontal lobe from the temporal lobe. The temporal lobe overlaps the insula, an island of cortex that is otherwise hidden, figure 8 to 19. The more posterior parieto-occipital sulcus separates the parietal lobe from the occipital lobe, figure 8-16c. In each lobe, some regions are concerned with sensory information and others with motor commands. Additionally, each hemisphere receives sensory information from, and sends motor commands to, the opposite side of the body. As a result, the left cerebral hemisphere controls the right side of the body, and the right cerebral hemisphere controls the left side. This crossing over has no known functional significance. Motor and sensory areas of the cortex. The major motor and sensory regions of the cerebral cortex are shown in figure 8 to 19. The central sulcus separates the motor and sensory portions of the cortex. The percentral gyrus of the frontal lobe forms the anterior margin of the central sulcus. The surface of this gyrus is the primary motor cortex. Neurons of the primary motor cortex direct voluntary movements by controlling somatic motor neurons in the brainstem and spinal cord. The post-central gyrus of the parietal lobe forms the posterior margin of the central sulcus. Its surface contains the primary somatosensory cortex. Neurons in this region receive somatic sensory information from touch, pressure, pain, and temperature receptors. We are aware of these sensations only when nuclei in the thalamus relay the information to the primary somatosensory cortex. Sensations of sight, taste, sound, and smell arrive at other portions of the cerebral cortex. The visual cortex of the occipital lobe receives visual information. The gustatory cortex of the frontal lobe receives taste sensations. In the temporal lobe, the auditory cortex and olfactory cortex receive information about hearing and smell, respectively. Association areas. The sensory and motor regions of the cortex are connected to nearby association areas. These regions interpret incoming data or coordinate a motor response. The somatosensory association area monitors activity in the primary somatosensory cortex. This area allows you to recognize a touch as light as a mosquito landing on your arm. The special senses of smell, sight, and hearing involve separate areas of sensory cortex. Each has its own association area. The premotor cortex, or somatic motor association area, is responsible for coordinating learned movements. When you perform a voluntary movement, such as picking up a glass or scanning these lines of type, instructions are relayed to the primary motor cortex by the premotor cortex. The functional distinctions between the motor and sensory association areas are most evident after localized brain damage has taken place. For example, someone with damage to the premotor cortex might understand written letters and words but be unable to read due to an inability to track along the lines on a printed page. In contrast, someone with a damaged visual association area can scan the lines of a printed page but cannot figure out what the letters mean. Cortical connections. The various regions of the cerebral cortex are interconnected by the white matter that lies beneath the cerebral cortex. Myelinated axons of different lengths interconnect gyri within a single cerebral hemisphere. Such axons also link the two hemispheres across the corpus callosum, figure 8-16c. Other bundles of axons link the cerebral cortex with the diencephalon, brainstem, cerebellum, and spinal cord. Cerebral processing centers. Higher order integrative centers receive information from many different association areas. These integrative centers direct extremely complex motor activities and perform complicated analytical functions. Even though integrative centers may be found in both cerebral hemispheres, many are lateralized, largely restricted to either the left or the right hemisphere, figure 8 to 20. Examples of lateralized integrative centers are those concerned with complex processes such as speech, writing, mathematical computation, and understanding spatial relationships. Figure 8 to 20 hemisphere lateralization. Functional differences in the left and right hemispheres of the brain. Some functional differences between the left and right cerebral hemispheres are depicted. Figure 8 to 20 full alternative text. The specialized language areas in the brain. Two important cortical areas related to human language are Wernicke's area and Broca's area. Both are primarily associated with the left cerebral hemisphere. Wernicke's area is near the auditory cortex and is associated with language comprehension. This analytical center receives information from the sensory association areas and plays an important role in your personality by integrating sensory information and coordinating access to visual and auditory memories. Damage to this area affects the ability to interpret what is read or heard, even though the words are understood as individual entities. For example, you might still understand the meaning of the spoken word sit and hear, because word recognition takes place in the auditory association areas. But you would be totally bewildered by the request sit here. Broca's area, motor speech area, also called the speech center, is near the motor cortex and is associated with speech production. Broca's area regulates the patterns of breathing and vocalization needed for normal speech. A person with damage to this area can make sounds but not words. The motor commands issued by the speech center are adjusted by feedback from the auditory association area. Damage there can cause a variety of speech-related problems. Some affected individuals have difficulty speaking, even though they know exactly which words to use. Others talk constantly but use all the wrong words. The prefrontal cortex. The prefrontal cortex of the frontal lobe, figure 8 to 20, coordinates information from the association areas of the entire cortex. In doing so, it performs such abstract intellectual functions as predicting the consequences of events or actions. Damage to this area leads to problems in estimating time relationships between events. Questions such as how long ago did this happen, or what happened first, become difficult to answer. The prefrontal cortex also has connections with other cortical areas and with other portions of the brain. Feelings of frustration, tension, and anxiety are generated at the prefrontal cortex as it interprets ongoing events and makes predictions about future situations or consequences. If the connections between the prefrontal cortex and other brain regions are severed, the tensions, frustrations, and anxieties are removed. Early in the 1900s, this rather drastic procedure, called a prefrontal lobotomy, was used to cure a variety of mental illnesses, especially those associated with violent or antisocial behavior. Hemispheric lateralization. As shown in figure 8 to 20, each of the two cerebral hemispheres is responsible for specific functions that are not ordinarily performed by the opposite hemisphere. This specialization is called hemispheric lateralization. In most people, the left hemisphere contains Wernicke's and Broca's areas and is responsible for language-based skills, reading, writing, and speaking. In addition, the premotor cortex involved in the control of hand movements is larger on the left side in right-handed individuals than in left-handed ones. The left hemisphere is also important in performing analytical tasks, such as mathematical calculations and logical decision-making. For these reasons, the left hemisphere has been called the dominant hemisphere, or the categorical hemisphere. The right hemisphere, or representational hemisphere, is concerned with spatial relationships and analyses. It analyzes sensory information and relates the body to the sensory environment. Interpretive centers in this hemisphere enable you to identify familiar objects by touch, smell, sight, or taste. The right hemisphere plays a dominant role in recognizing faces and in understanding three-dimensional relationships. It is also important in analyzing the emotional context of the conversation, for instance, distinguishing between the threat "get lost" and the question "get lost." Interestingly, there may be a link between handedness and sensory/spatial abilities. An unusually high percentage of musicians and artists are left-handed. The complex motor activities that they perform are directed by the primary motor cortex and association areas of the right cerebral hemisphere. These areas are near the association areas involved with spatial visualization and emotions. Hemispheric lateralization does not mean that the two hemispheres function independently of each other. Recall that the white fibers of the corpus callosum link the two hemispheres, including their sensory information and motor commands. The corpus callosum alone contains over 200 million axons, carrying an estimated 4 billion impulses per second. The electroencephalogram, EEG. Various methods have been used to map brain activity and function. The primary sensory cortex and the primary motor cortex have been mapped by direct stimulation in patients undergoing brain surgery. The behavioral changes that follow localized brain injuries or strokes can reveal the functions of other regions of the cerebrum. The activities of specific regions can be examined by non-invasive techniques such as a PET scan or sequential MRI scans. B20. The electrical activity of the brain is commonly monitored to assess brain activity. Neural function depends on electrical events of the plasma membrane of neurons. The brain contains billions of nerve cells, and their activity generates an electrical field that can be measured by placing electrodes on the brain or on the outer surface of the skull. The electrical activity changes constantly as nuclei and cortical areas are stimulated or quiet down. An electroencephalogram, EEG, is a printed record of this electrical activity over time. The electrical patterns are called brain waves, which can be correlated with the individual's level of consciousness. Electroencephalograms can also provide useful diagnostic information regarding brain disorders. 4 types of brainwave patterns are shown in figure 8 to 21. Figure 8 to 21 brain waves. 4 types of brainwave patterns and their descriptions. Figure 8 to 21 full alternative text. Clinical note: aphasia and dyslexia. Aphasia, A- without plus phasia, speech, is a disorder affecting the ability to speak or read. Global aphasia results from extensive damage to the general interpretive area or to the associated sensory tracts. Affected individuals cannot speak, read, understand, or interpret the speech of others. Global aphasia often accompanies a severe stroke or tumor that affects a large area of cortex, including the speech and language areas. Recovery is possible when the condition results from edema, an abnormal accumulation of fluid, or hemorrhage, but the process often takes months or even years. Lesser degrees of aphasia often follow minor strokes with no initial period of global aphasia. Such individuals can understand spoken and written words and may recover completely. Dyslexia, lexis, diction, is a disorder affecting the comprehension and use of words. Developmental dyslexia affects children. Estimates are that up to 15% of children in the United States suffer from some degree of dyslexia. These children have difficulty reading and writing, although their other intellectual functions may be normal or above normal. Their writing looks uneven and unorganized. They typically write letters in the wrong order. Dig becomes get, or reverse them, he becomes. Recent evidence suggests that at least some forms of dyslexia result from problems in processing, sorting, and integrating visual or auditory information. Jumbled letters of the alphabet. The word, dyslexia, is shown with the SL and E letters presenting backwards. Memory. What was the topic of the last sentence you read? What is your social security number? How do you open a screw top jar? To answer these questions, you access memories, stored bits of information gathered through experience. Fact memories are specific bits of information, such as the color of a stop sign. Skill memories are learned motor behaviors. You can probably remember how to light a match or throw a frisbee, for example. With repetition, skill memories become incorporated at the unconscious level. Examples include the complex motor patterns involved in skiing or playing the violin. Skill memories related to programmed behaviors, such as eating, are stored in appropriate portions of the brainstem. Complex skill memories involve an integration of motor patterns in the cerebellum and the cerebral cortex. Memories are often classified according to how long they last. Short-term memories do not last long, but while they persist, the information can be recalled immediately. Short-term memories contain small bits of information, such as a person's name or a telephone number. Repeating a phone number or other bit of information reinforces the original short-term memory and helps ensure its conversion to a long-term memory. Long-term memories remain for much longer periods, in some cases for an entire lifetime. The conversion from short-term to long-term memory is called memory consolidation. Some long-term memories fade with time and may require considerable effort to recall. Other long-term memories seem to be part of consciousness, such as your name or the contours of your own body. Most long-term memories are stored in the cerebral cortex. Conscious motor and sensory memories are referred to the appropriate association areas. For example, visual memories are stored in the visual association area, and memories of voluntary motor activity are kept in the premotor cortex. Special portions of the occipital and temporal lobes retain the memories of faces, voices, and words. Amnesia refers to the loss of memory as a result of disease or trauma. The type of memory loss depends on the specific regions of the brain affected. For example, damage to the auditory association areas may make it difficult to remember sounds. Damage to thalamic and limbic structures, especially the hippocampus, affects memory storage and consolidation. The basal nuclei. While your cerebral cortex is consciously active, other centers of your cerebrum, diencephalon, and brainstem are processing sensory information and issuing motor commands outside your conscious awareness. Many of these activities, which take place at the subconscious level, are directed by cerebral nuclei called basal nuclei. The basal nuclei are masses of gray matter deep within the brain. They lie beneath the lateral ventricles, surrounded by the white matter of each cerebral hemisphere. Figure 8 to 22. Figure 8 to 22. The basal nuclei. The relative positions of basal nuclei in the brain. Figure 8 to 22. Full alternative text. The caudate nucleus has a massive head and slender, curving tail that follows the curve of the lateral ventricle. The head of the caudate nucleus lies anterior to the lentiform, lens-shaped, nucleus. The lentiform nucleus consists of a medial globus pallidus, globus pallidus, pale globe, and a lateral putamen, putamen. Together, the caudate and lentiform nuclei are also called the corpus striatum, striated body. Inferior to the caudate and lentiform nuclei is another nucleus, the amygdaloid, amygdala, almond, body, or amygdala. It is a component of the limbic system and is discussed in the next section. Clinical note: seizures. A seizure is a temporary cerebral disorder accompanied by abnormal involuntary movements, unusual sensations, or inappropriate behavior. Clinical conditions characterized by seizures are known as seizure disorders, or epilepsies. Seizures of all kinds are accompanied by a marked change in the pattern of electrical activity seen in an electroencephalogram. The alteration begins in one portion of the cerebral cortex but may then spread across the entire cortical surface, like a wave on the surface of a pond. The nature of the signs and symptoms produced depends on the region of the cortex involved. If the seizure affects the primary motor cortex, the person will make involuntary movements. If it affects the auditory cortex, the person will hear strange sounds. The basal nuclei function in the subconscious control of skeletal muscle tone and the coordination of learned movement patterns. These nuclei do not start a movement. That decision is a voluntary one, but once a movement is underway, the basal nuclei provide the general pattern and rhythm. For example, when you walk, the basal nuclei control the cycles of arm and thigh movements that occur between the time you decide to start walking and the time you give the stop order. The limbic system. The limbic system, limbic, limbus, border, includes the olfactory cortex, several nuclei, gyri, and tracts along the border between the cerebrum and diencephalon, figure 8 to 23. This system is a functional grouping rather than an anatomical one. The functions of the limbic system include 1. Establishing emotional states, 2. Linking the conscious, intellectual functions of the cerebral cortex with the unconscious and autonomic functions of the brainstem, and 3. Aiding long-term memory storage and retrieval. In short, the sensory cortex, motor cortex, and association areas of the cerebral cortex enable you to perform complex tasks, but it is largely the limbic system that makes you want to do them. Figure 8 to 23. The limbic system. The relationship among the limbic system's major components. This three-dimensional reconstruction of the limbic system shows the relationships among the system's major components. Figure 8 to 23. Full alternative text. The amygdaloid bodies link the limbic system, the cerebrum, and various sensory systems. These cerebral nuclei play a role in regulating heart rate, responding to fear and anxiety, controlling the fight or flight response, and linking emotions with specific memories. Another nucleus, the hippocampus, is important in learning and in storing long-term memories. Damage to the hippocampus in Alzheimer's disease interferes with memory storage and retrieval. The fornix, arch, is attractive white matter that connects the hippocampus with the hypothalamus, figures 8-16c and 8 to 23. The limbic system also includes hypothalamic centers that control 1. Emotional states, such as rage, fear, and sexual arousal, and 2. Reflex movements that can be consciously activated. For example, the limbic system includes the mammillary bodies, mammillary, mamilla, a little breast, of the hypothalamus, where many fibers of the fornix end. These nuclei process olfactory sensations and control reflex movements associated with eating, such as chewing, licking, and swallowing. The diencephalon. The diencephalon, figures 8-16c and 8 to 24, contains switching and relay centers that integrate conscious and unconscious sensory information and motor commands. It surrounds the third ventricle and consists of the epithalamus, thalamus, and hypothalamus. The epithalamus. The epithalamus lies superior to the third ventricle, where it forms the roof of the diencephalon. The anterior portion contains an extensive area of choroid plexus. The posterior portion contains the pineal gland, figure 8-24b, an endocrine structure that secretes the hormone melatonin. Among other functions, melatonin is important in regulating day-night cycles. Figure 8 to 24. The diencephalon and brainstem. Structures of the diencephalon and brainstem. Figure 8 to 24. Full alternative text. The thalamus. The left thalamus and right thalamus are separated by the third ventricle. Each contains a rounded mass of thalamic nuclei. The thalamus, figures 8-16c and 8-24, is the final relay point for all ascending sensory information, other than olfactory, that will reach our conscious awareness. It acts as a filter, passing on to the primary sensory cortex only a small portion of the arriving sensory information. The rest is relayed to the basal nuclei and centers in the brainstem. The thalamus also plays a role in coordinating voluntary and involuntary motor commands. The hypothalamus. The hypothalamus lies inferior to the third ventricle, figure 8-16c. The hypothalamus contains important control and integrative centers in addition to those associated with the limbic system. Its diverse functions include subconscious control of skeletal muscle contractions associated with rage, pleasure, pain, and sexual arousal. Adjusting the activities of autonomic centers in the pons and medulla oblongata, such as heart rate, blood pressure, respiration, and digestive functions. Coordinating activities of the nervous and endocrine systems. Secreting a variety of hormones, including antidiuretic hormone, ADH, and oxytocin, OXT. Producing the behavioral drives involved in hunger and thirst. Coordinating voluntary and autonomic functions. Regulating normal body temperature, and coordinating the daily cycles of activity. The midbrain. The midbrain, figure 8-24, contains various nuclei and bundles of ascending and descending nerve fibers. It includes two pairs of sensory nuclei, or colliculi, caoliculi, singular, colliculus, a small hill, involved in processing visual and auditory sensations. The superior colliculi control the reflex movements of the eyes, head, and neck in response to visual stimuli, such as a bright light. The inferior colliculi control reflex movements of the head, neck, and trunk in response to auditory stimuli, such as a loud noise. The midbrain also contains motor nuclei for two of the cranial nerves, 3, 4, involved in the control of eye movements. Descending bundles of nerve fibers on the ventrolateral surface of the midbrain make up the cerebral peduncles, little feet. Some of the descending fibers go to the cerebellum by way of the pons. Others carry voluntary motor commands from the primary motor cortex of each cerebral hemisphere. The midbrain is also headquarters to one of the most important brainstem components, the reticular formation, which regulates many involuntary functions. The reticular formation is a network of interconnected nuclei that extends the length of the brainstem. The reticular formation of the midbrain contains the reticular activating system, RAS. The output of the system directly affects the activity of the cerebral cortex. When the RAS is inactive, so are we. When the RAS is stimulated, so is our state of attention or wakefulness. Midbrain nuclei that integrate information from the cerebrum and cerebellum control the maintenance of muscle tone and posture. Other midbrain nuclei play an important role in regulating the motor output of the basal nuclei. For example, the substantia nigra, N-nigrou, black, inhibit the activity of the basal nuclei by releasing the neurotransmitter dopamine. P260 if the substantia nigra are damaged or the neurons secrete less dopamine, the basal nuclei become more active. The result is a gradual increase in muscle tone and the appearance of symptoms characteristic of Parkinson's disease. Persons with Parkinson's disease have difficulty starting voluntary movements because opposing muscle groups do not relax. Instead, they must be overpowered. Once a movement is underway, every aspect must be voluntarily controlled through intense effort and concentration. The pons. The pons, figure 8-24a, links the cerebellum with the midbrain, diencephalon, cerebrum, and spinal cord. One group of nuclei within the pons includes the sensory and motor nuclei for 4 of the cranial nerves, V, A. Other nuclei are concerned with the involuntary control of the pace and depth of respiration. Tracks passing through the pons link the cerebellum with the brainstem, cerebrum, and spinal cord. The cerebellum. The cerebellum, figure 8-16bc, is an automatic processing center. Like the cerebrum, the cerebellum is composed of white matter covered by a layer of neural cortex, gray matter, called the cerebellar cortex. The cerebellum has two important functions. Adjusting the postural muscles of the body. The cerebellum coordinates rapid, automatic adjustments to maintain balance and equilibrium. Programming and fine-tuning movements controlled at the conscious and subconscious levels. The cerebellum refines learned movement patterns, such as riding a bicycle or playing the piano. These functions are performed indirectly by regulating activity along motor pathways at the cerebral cortex, basal nuclei, and brainstem. The cerebellum compares the motor commands with proprioceptive information, position sense, and makes adjustments needed to make the movement smooth. The tracks that link the cerebellum with these different regions are the cerebellar peduncles, figure 8-24. The cerebellum can be permanently damaged by trauma or stroke or temporarily affected by drugs such as alcohol. These alterations can produce ataxia, ataxia, a lack of order, a disturbance in balance. The medulla oblongata. The medulla oblongata, figure 8-24, connects the brain with the spinal cord. It is a very busy place. All communication between the brain and spinal cord involves tracts that ascend or descend through the medulla oblongata. These tracts often synapse in the medulla oblongata at sensory or motor nuclei that act as relay stations and processing centers. In addition to these nuclei, the medulla oblongata contains sensory and motor nuclei associated with 5 of the cranial nerves, 8, 12. The portion of the reticular system within the medulla oblongata contains nuclei and centers that regulate vital autonomic functions. These reflex centers receive inputs from cranial nerves, the cerebral cortex, and the brainstem. Their output controls or adjusts the activities of the cardiovascular and respiratory systems. The cardiovascular center adjusts heart rate, the strength of cardiac contractions, and the flow of blood through peripheral tissues. In terms of function, the cardiovascular center is subdivided into a cardiac center regulating the heart rate and a vasomotor center controlling peripheral blood flow. The respiratory rhythmicity centers set the basic pace for respiratory movements, and their activity is adjusted by the respiratory centers of the pons. Checkpoint. Describe one major function of each of the 6 regions of the brain. The pituitary gland links the nervous and endocrine systems. To which portion of the diencephalon is it attached? How would decreased diffusion across the arachnoid granulations affect the volume of cerebrospinal fluid in the ventricles? Mary suffers a head injury that damages her primary motor cortex. Where is this area located? Which senses would be affected by damage to the temporal lobes of the cerebrum? The thalamus acts as a relay point for all but what type of sensory information? Changes in body temperature stimulate which area of the diencephalon? The medulla oblongata is one of the smallest sections of the brain. Why can damage to it cause death, when similar damage in the cerebrum might go unnoticed? See the Blue Answers tab at the back of the book. 8 to 8 The PNs connects the CNS with the body's external and internal environments. Learning outcome. Name the cranial nerves, relate each pair of cranial nerves to its principal functions, and relate the distribution pattern of spinal nerves to the regions they innervate. The peripheral nervous system, PNs, links the neurons of the central nervous system, CNS, to the rest of the body. All sensory information and motor commands are carried by axons of the PNs, figure 8 to 1, P245. These axons are bundled together and wrapped in connective tissue, forming peripheral nerves, or simply nerves. Cranial nerves originate from the brain, and spinal nerves connect to the spinal cord. The PNs also includes both the cell bodies and the axons of sensory neurons and motor neurons of the autonomic nervous system. The cranial nerves. 12 pairs of cranial nerves connect to the brain, figure 8 to 25. Each cranial nerve has a name related to its distribution or function. Each also has a designation consisting of the letter CN and a Roman numeral, or its position along the longitudinal axis of the brain. For example, CN2 refers to the second pair of cranial nerves, the optic nerves. If the full name of the cranial nerve is given, then only the Roman numeral is needed, such as optic nerve, 2. Figure 8 to 25 The cranial nerves. In the brain, the cranial nerves, their functions, and innervation are identified. Figure 8 to 25 Full alternative text. Distribution and function of cranial nerves. Functionally, each nerve can be classified as primarily sensory, primarily motor, or mixed, sensory and motor. Many cranial nerves, however, have secondary functions. For example, several cranial nerves, 3, 7, 9, and X, also carry autonomic fibers to PNs ganglia, just as spinal nerves deliver them to ganglia along the spinal cord. Next we consider the distribution and functions of the cranial nerves. Few people are able to remember the names, numbers, and functions of the cranial nerves without some effort. Many people use mnemonic phrases, such as, oh, once one takes the anatomy final, very good vacations are heavenly. The first letter of each word represents the name of a cranial nerve. The olfactory nerves, I. The first pair of cranial nerves, the olfactory nerves, are the only cranial nerves attached to the cerebrum. The rest start or end with a nuclei of the diencephalon or brainstem. These nerves carry special sensory information responsible for the sense of smell. The olfactory nerves originate in the epithelium of the upper nasal cavity and penetrate the cribriform plate of the ethmoid bone to synapse in the olfactory bulbs of the brain. P157 From the olfactory bulbs, the axons of postsynaptic neurons travel within the olfactory tracts to the olfactory centers of the brain. The optic nerves, 2. The optic nerves carry visual information from the eyes. After passing through the optic foramina of the orbits, these nerves intersect at the optic chiasm, a crossing, figure 8-25AB, before they continue as the optic tracts to nuclei of the left and right thalamus. The oculomotor nerves, 3. The midbrain contains the motor nuclei controlling the third and fourth cranial nerves. Each oculomotor nerve innervates 4 of the 6 extrinsic muscles that move an eyeball, the superior, medial, and inferior rectus muscles and the inferior oblique muscle. Superficial muscles that position or stabilize an organ are called extrinsic muscles. These nerves also carry autonomic fibers to intrinsic eye muscles that control the amount of light entering the eye and the shape of the lens. The muscles located entirely within an organ are called intrinsic muscles. The trochlear nerves, 4. The trochlear, Troclear, trochlea, a pulley, nerves, the smallest of the cranial nerves, innervate the superior oblique muscles of the eyes. The motor nuclei that control these nerves lie in the midbrain. The name trochlear refers to the pulley-shaped, ligamentous link through which the tendon of the superior oblique muscle passes to reach its attachment on the eyeball, figure 9-9A, P316. The trigeminal nerves, V. The pons contains the nuclei associated with cranial nerve V. The trigeminal, trigeminal, nerves are the largest of the cranial nerves. These nerves provide sensory information from the head and face and motor control over the chewing muscles, such as the temporalis and masseter. The trigeminal has 3 major divisions. The ophthalmic nerve, V1, provides sensory information from the orbit of the eye, the nasal cavity and sinuses, and the skin of the forehead, eyebrows, eyelids, and nose. The maxillary nerve, V2, provides sensory information from the lower eyelid, upper lip, cheek, nose, upper gums, and teeth, palate, and portions of the pharynx. The mandibular nerve, V3, is the largest of the 3. It provides sensory information from the skin of the temples, the lower gums and teeth, the salivary glands, and the anterior portions of the tongue. It also provides motor control over the chewing muscles, the temporalis, masseter, and pterygoid muscles. B. 219. The abducens nerves, 6. The abducens nerves innervate only the lateral rectus, the 6th of the extrinsic eye muscles. The nuclei of the abducens nerves are in the pons. The nerves emerge at the border between the pons and the medulla oblongata and reach the orbit of the eye along with the oculomotor and trochlear nerves. The name abducens is based on the action of this nerve's innervat muscle, which abducts the eyeball, causing it to rotate laterally, away from the midline of the body. The facial nerves, 7. The facial nerves are mixed nerves of the face. Their sensory and motor roots emerge from the side of the pons. The sensory fibers monitor proprioceptors in the facial muscles, provide deep pressure sensations over the face, and provide taste information from receptors along the anterior 2/3 of the tongue. The motor fibers produce facial expressions by controlling the superficial muscles of the scalp and face and muscles near the ear. These nerves also carry autonomic fibers that result in control of the tear glands and salivary glands. The vestibulocochlear nerves, 8. The vestibulocochlear nerves monitor the sensory receptors of the internal ear. The pons and medulla oblongata contain nuclei associated with these nerves. Each vestibulocochlear nerve has two parts. 1. A vestibular nerve, vestibulum, a cavity, which originates at the vestibule, the portion of the internal ear concerned with balance sensations, and conveys information on position, movement, and balance. And 2. The cochlear, cochlea, snail shell, nerve, which monitors the receptors of the cochlea, the portion of the internal ear responsible for the sense of hearing. The glossopharyngeal nerves. 9. The glossopharyngeal, glossopharyngeal, glossus, tongue, nerves are mixed nerves innervating the tongue and pharynx. The associated sensory and motor nuclei are in the medulla oblongata. The sensory portion of this nerve provides taste sensations from the posterior third of the tongue. It also monitors blood pressure and dissolved gas concentrations in major blood vessels. The motor portion controls the pharyngeal muscles involved in swallowing. These nerves also carry autonomic fibers that control the parotid salivary glands. The vagus nerves, X. The vagus, wandering, nerves provide sensory information from each auricle and external acoustic meatus, auditory canal, the diaphragm, and taste receptors in the pharynx, and from visceral receptors along the esophagus, respiratory tract, and abdominal organs as far away as the last portions of the large intestine. The associated sensory and motor nuclei of the vagus are located in the medulla oblongata. The sensory information that is provided is vital to the autonomic control of visceral function. We are not consciously aware of these sensations because they are seldom related to the cerebral cortex. The motor components of the vagus nerves control skeletal muscles of the soft palate, pharynx, and esophagus and affect cardiac muscle, smooth muscle, and glands of the esophagus, stomach, intestines, and gallbladder. The accessory nerves, 11. The accessory nerves, sometimes called the spinal accessory nerves, are motor nerves that innervate structures in the neck and back. These nerves differ from other cranial nerves in that some of their motor fibers originate in the lateral gray horns of the first 5 cervical segments of the spinal cord, as well as in the medulla oblongata. All these motor fibers join together in the cranium and exit as cranial nerve 11, which then divides into 2 branches. The internal branch joins the vagus nerve and innervates the voluntary swallowing muscles of the soft palate and pharynx, as well as the laryngeal muscles that control the vocal cords and produce speech. The external branch controls the sternocleidomastoid and trapezius muscles associated with the pectoral girdles. Pages 220. 225. The hypoglossal nerves, 12. The hypoglossal nerves provide voluntary control over movements of the tongue. The nuclei for these motor nerves are located in the medulla oblongata. The distribution, innervat structures, and functions of the cranial nerves are summarized in Figure 8-25b. The spinal nerves. The 31 pairs of spinal nerves are grouped according to the region of the vertebral column from which they originate. Figure 8-26. They include 8 pairs of cervical nerves, C1, C8, 12 pairs of thoracic nerves, T1, T12, 5 pairs of lumbar nerves, L1, L5, 5 pairs of sacral nerves, S1, S5, and 1 pair of coccygeal nerves, CO1. Figure 8-26 peripheral nerves and nerve plexuses. The 4 spinal nerve plexuses and major peripheral nerve distributions are shown. Figure 8-26 full alternative text. Each pair of spinal nerves monitors a specific region of the body surface known as a dermatome. Figure 8-27. Dermatomes are clinically important because damage or infection of a spinal nerve or of spinal ganglia produces a characteristic loss of sensation in the corresponding region of the skin. For example, in shingles, a virus that infects spinal ganglia causes a painful rash whose distribution corresponds to that of the affected sensory nerves. Figure 8-27 dermatomes. Locations of dermatomes on the surface of the skin are shown in anterior and posterior view. Distributions of dermatomes on the surface of the skin, as seen in the anterior and posterior view. The face is served by cranial nerves, not spinal nerves. Figure 8-27 full alternative text. Nerve plexuses. During development, skeletal muscles commonly fuse, forming larger muscles innervat by nerve trunks containing axons derived from several spinal nerves. These compound nerve trunks originate at networks called nerve plexuses. The 4 spinal nerve plexuses and the distribution of some of the major peripheral nerves are shown in Figure 8-26. The cervical plexus innervates the muscles of the neck and extends into the thoracic cavity to control the diaphragm, a key respiratory muscle. The brachial plexus innervates the pectoral girdles and upper limbs. The lumbar plexus and the sacral plexus supply the pelvic girdle and lower limbs. These plexuses are sometimes designated the lumbosacral plexus. The nerves arising at the nerve plexuses contain sensory as well as motor fibers. Peripheral nerve palsies, also known as peripheral neuropathies, are characterized by regional losses of sensory and motor function as the result of nerve trauma or compression. You have experienced a mild, temporary palsy if your arm or leg has ever fallen asleep. Checkpoint. What signs would you associate with damage to the abducens nerve? 6. John is having trouble moving his tongue. His physician tells him it is due to pressure on a cranial nerve. Which cranial nerve is involved? Injury to which nerve plexus would interfere with the ability to breathe? See the blue answers tab at the back of the book. 8 to 9 reflexes are rapid, automatic responses to stimuli. Learning outcome. Describe the steps in a reflex arc. We can study the central and peripheral nervous systems separately, but they work together. To consider the ways the CNS and PNs interact, we begin with simple reflex responses to stimulation. A reflex is a rapid, automatic response to a specific stimulus. Reflexes help preserve homeostasis by making rapid adjustments in the function of organs or organ systems. The response shows little variability. Whenever a particular reflex is activated, it usually produces the same motor response. Simple reflexes. A reflex involves sensory fibers delivering information from peripheral receptors to the CNS, and motor fibers carrying motor commands to peripheral effectors. The wiring of a single reflex is called a reflex arc. It begins at a receptor and ends at an effector such as a muscle fiber or gland cell. Figure 8-28 diagrams the 5 steps involved in the action of a reflex arc. 1. The arrival of a stimulus and activation of a receptor. 2. The activation of a sensory neuron. 3. Information processing by an interneuron. 4. The activation of a motor neuron. And 5. The response by a peripheral effector. Figure 8-28 events in a reflex arc. The 5 steps that occur in a reflex arc. Figure 8-28 full alternative text. A reflex usually removes or opposes the original stimulus. In Figure 8-28, the contracting muscle pulls the hand away from the painful stimulus. This reflex arc is, therefore, an example of negative feedback. P10 by opposing potentially harmful changes in the internal or external environment, reflexes play an important role in maintaining homeostasis. In the simplest reflex arc, a sensory neuron synapses directly on a motor neuron, which performs the information processing function. Such a reflex is called a monosynaptic reflex. Because there is only one synapse, monosynaptic reflexes control the most rapid, stereotyped motor responses of the nervous system. The best known example is the stretch reflex. The stretch reflex provides automatic regulation of skeletal muscle length. The sensory receptors in the stretch reflex are called muscle spindles. They are bundles of small, specialized skeletal muscle fibers scattered throughout skeletal muscles. The stimulus, increasing muscle length, activates a sensory neuron that triggers an immediate motor response, contraction of the stretched muscle, that counteracts the stimulus. Stimulus. Stretch reflexes are important in maintaining normal posture and balance and in making automatic adjustments in muscle tone. Physicians can use the sensitivity of a stretch reflex to test the general condition of the spinal cord, peripheral nerves, and muscles. For example, in the patellar reflex, or knee-jerk reflex, a sharp wrap on the patellar tendon stretches muscle spindles in the quadriceps muscles, figure 8-29. With so brief a stimulus, the reflexive contraction occurs unopposed and produces a noticeable kick. If this contraction shortens the muscle spindles to less than their original resting lengths, the sensory nerve endings are compressed, the sensory neuron is inhibited, and the leg drops back. Figure 8-29A stretch reflex. The 2 steps that occur in the patellar reflex. The patellar reflex is a stretch reflex controlled by stretch receptors, muscle spindles, and the quadriceps muscles that straighten the knee. When a reflex hammer strikes the patellar tendon, the muscle spindles are stretched. This stretching results in a sudden increase in the activity of the sensory neurons, which synapse on motor neurons in the spinal cord. The activation of spinal motor neurons produces an immediate muscle contraction and a reflexive kick. Figure 8-29 full alternative text. Complex reflexes. Many spinal reflexes have at least one interneuron between the sensory, afferent, neuron and the motor, efferent, neuron, figure 8-28. Because there are more synapses, such polysynaptic reflexes include a longer delay between stimulus and response. But they can produce far more responses because the interneurons can control several muscle groups simultaneously. Withdrawal reflexes move stimulated parts of the body away from a source of stimulation. Painful stimuli trigger the strongest withdrawal reflexes, but these reflexes are also initiated by the stimulation of touch or pressure receptors. A flexor reflex is a withdrawal reflex affecting the muscles of a limb. If you grab an unexpectedly hot pan on the stove, a dramatic flexor reflex occurs, figure 8-30. When the pain receptors in your hand are stimulated, the sensory neurons activate interneurons in the spinal cord that stimulate motor neurons in the anterior gray horns. As a result, flexor muscles contract and yank your forearm and hand away from the stove. At the same time this response is taking place, pain sensations are ascending to the brain. Figure 8-30 the flexor reflex, a type of withdrawal reflex. The 3 steps that occur in the flexor reflex of the hand. Figure 8-30 full alternative text. When a specific muscle contracts, opposing, antagonistic muscles are stretched. For example, the flexor muscles that bend the elbow are opposed by extensor muscles, which straighten it out. A potential conflict exists here. Contraction of a flexor muscle should trigger a stretch reflex in the extensors that would cause them to contract, opposing the movement that is underway. Interneurons in the spinal cord prevent such competition through reciprocal inhibition. When one set of motor neurons is stimulated, those controlling antagonistic muscles are inhibited. Integration and control of spinal reflexes. Reflexes are automatic, but higher centers in the brain influence these responses. They do so by facilitating, assisting, or inhibiting the interneurons and motor neurons involved. The sensitivity of a reflex can thus be modified. The facilitation of motor neurons involved in reflexes is called reinforcement. For example, a method used to overemphasize the patellar reflex is a distractive technique called the Androsic maneuver. To do this, the person hooks the hands together by interlocking the fingers and then tries to pull the hands apart while a light tap is applied to the patellar tendon. This reinforcement produces a big kick rather than a twitch. A larger reflex response occurs even if the individual realizes it is just a distraction. Other descending fibers have an inhibitory effect on spinal reflexes. Stroking an infant's foot on the side of the sole produces a fanning of the toes known as the Babinski reflex. This response disappears as descending inhibitory synapses develop, so in adults the same stimulus produces a curling of the toes, called the plantar reflex, or negative Babinski reflex, after about a 1-second delay. If either the higher centers or the descending tracts are damaged, the Babinski sign will reappear. As a result, clinicians often test this reflex if CNS injury is suspected. Spinal reflexes produce consistent, stereotyped motor patterns that are triggered by specific external stimuli. However, the same motor patterns can also be activated as needed by higher centers in the brain. The use of pre-existing motor patterns allows a relatively small number of descending fibers to control complex motor functions. For example, the motor patterns for walking, running, and jumping are directed primarily by neuronal pools in the spinal cord. The descending pathways from the brain facilitate, inhibit, or fine-tune the established patterns. Checkpoint. Define reflex. Which common reflex do physicians use to test the general condition of the spinal cord, peripheral nerves, and muscles? Why do polysynaptic reflexes produce more complex responses than monosynaptic reflexes? After injuring his back lifting a sofa, Tom exhibits a positive Babinski reflex. What does this imply about Tom's injury? See the Blue Answers tab at the back of the book. Build your knowledge. Recall that sensory receptors are present in the dermis and deeper layers of the epidermis, as you saw in Chapter 5, the integumentary system. Dermal nerve fibers monitor these sensory receptors, which provide sensations of touch, pain, pressure, and temperature. 129. 8 to 10 separate pathways carry sensory information and motor commands. Learning outcome. Identify the principal sensory and motor pathways, and explain how it is possible to distinguish among sensations that originate in different areas of the body. The communication among the CNS, the PNs, and organ systems takes place over pathways, nerve tracts, and nuclei that relay sensory and motor information. P250, the names of the major sensory, ascending, and motor, descending, tracts of the spinal cord are based on the destinations of the axons. If the name of the tract begins with spino dash, the tract starts in the spinal cord and ends in the brain, and it therefore carries sensory information. If the name of the tract ends in spinal, its axons start in the higher centers and end in the spinal cord, bearing motor commands. The rest of the tract's name indicates the associated nucleus or cortical area of the brain. Table 8.1 lists some examples of sensory and motor pathways, and their functions. Table 8.1 Sensory and motor pathways. Sensory pathways. Sensory receptors monitor conditions in the body or the external environment. The information gathered by a sensory receptor arrives in the CNS in the form of action potentials in an afferent, sensory, fiber. The arriving information is called a sensation. Most processing of arriving sensations takes place in centers along the sensory pathways in the spinal cord or brainstem. Only about 1% of the arriving information reaches the cerebral cortex and our conscious awareness. For example, we usually do not perceive, or feel, the clothes we wear or hear the hum of our car's engine. The posterior column pathway. One example of an ascending sensory pathway is the posterior column pathway, figure 8 to 31. It sends highly localized, fine, touch, pressure, vibration, and proprioception, position, sensations to the cerebral cortex. In the process, the information is relayed from one neuron to another. Figure 8 to 31, the posterior column pathway. The process of information traveling through the posterior column pathway. The posterior column pathway carries fine touch, pressure, vibration, and proprioception sensations to the primary somatosensory cortex of the cerebral hemisphere on the opposite side of the body. For clarity, this figure shows only the pathway for sensations originating on the right side of the body. Figure 8 to 31, full alternative text. Sensations travel along the axon of a sensory neuron, reaching the CNS through the dorsal roots of spinal nerves, in figure 8 to 31. Within the spinal cord, the axons ascend within the posterior column pathway. They synapse in the sensory nucleus of the medulla oblongata. The axons of the neurons in this nucleus, the second neuron in this pathway, cross over to the opposite side of the brainstem before continuing to the thalamus. The location of the synapse in the thalamus depends on the region of the body involved. The thalamic, in this case, third, neuron then relays the information to an appropriate region of the primary somatosensory cortex. The sensations arrive at particular locations in the primary somatosensory cortex. For example, sensory information from the toes reaches one end of the primary somatosensory cortex, and information from the head reaches the other. In effect, the primary somatosensory cortex contains a miniature map of the body surface called the sensory homunculus, little man. That map is distorted because the area of sensory cortex devoted to a particular region is proportional not to its size, but to the number of sensory receptors the region contains. In other words, it takes many more cortical neurons to process sensory information from the tongue, which has tens of thousands of taste and touch receptors, than it does to analyze sensations from the back, where touch receptors are few and far between. Motor pathways. In response to information from sensory systems, the CNS issues motor commands that are distributed by the somatic nervous system, SNS, and the autonomic nervous system, ANS, of the efferent division of the PNs, figure 8 to 1, P245. The SNS, under voluntary control, issues somatic motor commands that direct the contractions of skeletal muscles. The motor commands of the ANS occur outside our conscious awareness. They control the smooth muscles, cardiac muscle, and glands. Three motor pathways provide control over skeletal muscles, the corticospinal pathway, the medial pathway, and the lateral pathway. The corticospinal pathway provides conscious, voluntary control over skeletal muscles. The medial and lateral pathways XERT more indirect, subconscious control. Table 8 to 1 lists some examples and functions of these motor pathways. We begin our examination of motor pathways with the corticospinal pathway. The corticospinal pathway. The corticospinal pathway, sometimes called the pyramidal system, provides conscious, voluntary control of skeletal muscles. Figure 832 shows the motor pathway providing voluntary control over the right side of the body. As in the case of the sensory homunculus, in the primary motor cortex, the area devoted to a body part does not reflect the size of that part. Instead, the area reflects the number of motor units present in that part of the body. For example, the motor homunculus has grossly oversized hands. Their size is an indication of the large number of motor units involved in writing, grasping, and manipulating objects in our environment. Figure 8 to 32, the corticospinal pathway. The process of voluntary controls of skeletal muscles in the corticospinal pathway. The corticospinal pathway originates at the primary motor cortex. Axons of the pyramidal cells of the primary motor cortex descend to reach motor nuclei in the brainstem and spinal cord. Most of the fibers cross over in the medulla oblongata before descending into the spinal cord as the corticospinal tracts. Figure 8 to 32 full alternative text. The corticospinal pathway begins at triangular-shaped pyramidal cells of the cerebral cortex. The axons of these upper motor neurons extend into the brainstem and spinal cord, where they synapse on lower motor neurons, in figure 8 to 32. All axons of the corticospinal tracts eventually cross over to reach motor neurons on the opposite side of the body. As a result, the right cerebral hemisphere controls the left side of the body, and the left cerebral hemisphere controls the right side. The medial and lateral pathways. The medial and lateral pathways provide subconscious, involuntary control of muscle tone and movements of the neck, trunk, and limbs. They also coordinate learned movement patterns and other voluntary motor activities. Table 8 to 1. Together, these pathways were known as the extrapyramidal system because it was thought that they operated independently of and parallel to the pyramidal system, the corticospinal pathway. We now know that the control of the body's motor functions is integrated among all three motor pathways. The components of the medial and lateral pathways are spread throughout the brain. These components include nuclei in the brainstem, midbrain, pons, and medulla oblongata, relay stations in the thalamus, the basal nuclei of the cerebrum, and the cerebellum. Output from the basal nuclei and cerebellum exerts the highest level of control. For example, their output can stimulate or inhibit 1. Other nuclei of these pathways, or 2. The activities of pyramidal cells in the primary motor cortex. Also, axons from the upper motor neurons in the medial and lateral pathways synapse on the same motor neurons innervat by the corticospinal pathway. Clinical note: cerebral palsy. The term cerebral palsy refers to a number of disorders affecting voluntary motor performance, including speech, movement, balance, and posture, that appear during infancy or childhood and persist throughout life. The cause may be trauma associated with premature or unusually stressful birth, maternal exposure to drugs, including alcohol, or a genetic defect that causes the motor pathways to develop improperly. Problems with labor and delivery result from the compression or interruption of placental circulation or oxygen supplies. If the oxygen concentration of fetal blood declines significantly for as little as 5 to 10 minutes, CNS function can be permanently impaired. The cerebral cortex, cerebellum, basal nuclei, hippocampus, and thalamus are likely to be affected. Abnormalities in motor skills, posture, and balance, memory, speech, and learning abilities may result. Checkpoint. As a result of pressure on her spinal cord, Jill cannot feel touch or pressure on her legs. What sensory pathway is being compressed? The primary motor cortex of the right cerebral hemisphere controls motor function on which side of the body? An injury to the superior portion of the primary motor cortex would affect the ability to control muscles of which parts of the body? See the Blue Answers tab at the back of the book. 8 to 11. The autonomic nervous system, composed of the sympathetic and parasympathetic divisions, is involved in the unconscious regulation of body functions. Learning outcome. Describe the structures and functions of the sympathetic and parasympathetic divisions of the autonomic nervous system. Your conscious sensations, plans, and responses are only a tiny fraction of the activities of the nervous system. In practical terms, conscious activities have little to do with our immediate or long-term survival. The autonomic nervous system, ANS, makes adjustments that are much more important. Without the ANS, a simple night's sleep would be a life-threatening event. Recall that both the ANS and the somatic nervous system, SNS, are parts of the efferent division of the PNs. As such, they carry motor commands to peripheral effectors. 245, however, clear anatomical differences exist between them. Figure 8 to 33. In the SNS, lower motor neurons exert direct control over skeletal muscles. Figure 833. In the ANS, a second motor neuron always separates the CNS and the peripheral effector. Figure 833b. We now turn to the ANS. Figure 8 to 33, the organization of the somatic and autonomic nervous systems. The structures and organization of the somatic and autonomic nervous systems. Figure 8 to 33, full alternative text. The ANS motor neurons in the CNS are known as pregangliotic neurons. They send their axons, called pregangliotic fibers, to autonomic ganglia outside the CNS. In these ganglia, the axons of pregangliotic neurons synapse on gangliotic neurons. The axons of the gangliotic neurons are postgangliotic fibers. These fibers leave the ganglia and innervate cardiac muscle, smooth muscles, glands, and, in some cases, fat cells, adipocytes. The ANS has two divisions, the sympathetic division and the parasympathetic division. In the sympathetic division, pregangliotic fibers leave the thoracic and lumbar segments of the spinal cord and synapse in ganglia near the spinal cord, figure 8 to 34. In this division, the pregangliotic fibers are short, and the postgangliotic fibers are long. The sympathetic division is often called the fight or flight system because it usually stimulates tissue metabolism, increases alertness, and prepares the body to deal with emergencies. Figure 8 to 34, the sympathetic division. The distribution of sympathetic fibers in the brain, spinal cord, and various organs. The distribution of sympathetic fibers is the same on both sides of the body. For clarity, the innervation of somatic structures is shown to the left, and the innervation of visceral structures to the right. Figure 8 to 34, full alternative text. In the parasympathetic division of the ANS, pregangliotic fibers originate in the brainstem and the sacral segments of the spinal cord, figure 8 to 35. They synapse on neurons in terminal ganglia very close to the target organs, or in intramural ganglia, murus, wall, embedded within the target organs. In this division, the pregangliotic fibers are long, and the postgangliotic fibers are short. The parasympathetic division is often regarded as the rest and repose or rest and digest system because it conserves energy and promotes sedentary activities, such as digestion. Figure 8 to 35, the parasympathetic division. The distribution of parasympathetic fibers in the brain, spinal cord, and various organs. The distribution of parasympathetic fibers is the same on both sides of the body. Figure 8 to 35, full alternative text. Both divisions of the ANS affect target organs by releasing specific neurotransmitters from their postgangliotic fibers. The result may be either stimulation or inhibition of activity, depending on the membrane receptor's response to the neurotransmitter. Some general patterns are worth noting. All pregangliotic autonomic fibers are cholinergic. They release acetylcholine, ACH, at their axon terminals. P258, the effects are always excitatory. Postgangliotic parasympathetic fibers are also cholinergic. However, the effects are excitatory or inhibitory, depending on the nature of the target cell receptor. Most postgangliotic sympathetic fibers release norepinephrine, NE. Neurons that release NE are called adrenergic. P260, the effects of NE are usually excitatory. The sympathetic division. The sympathetic division of the ANS, figure 8 to 34, consists of the following components. Pregangliotic neurons located between segments T1 and L2 of the spinal cord. The cell bodies of these neurons are found in the lateral gray horns. Their short axons enter the anterior roots of these spinal segments. Gangliotic neurons located in ganglia near the vertebral column. Two types of sympathetic ganglia exist. Paired sympathetic chain ganglia lie on either side of the vertebral column. They contain neurons that control effectors in the body wall and inside the thoracic cavity. Unpaired collateral ganglia lie interior to the vertebral column. They contain gangliotic neurons that innervate tissues and organs in the abdominopelvic cavity. The adrenal medulla. The center of each adrenal, near plus renal, kidney, gland is known as the adrenal medulla. It is a modified sympathetic ganglion. Its gangliotic neurons have very short axons. Organization of the sympathetic division. Figure 834 diagrams the distribution of sympathetic innervation. Note that this distribution is the same on both sides of the body. The sympathetic chain. From spinal segments T1 to L2, sympathetic pregangliotic fibers join the ventral root of each spinal nerve. All these fibers then exit the spinal nerve to enter the sympathetic chain ganglia, figure 8 to 34. The fibers diverge extensively, with one pregangliotic fiber synapsing on 2 dozen or more gangliotic neurons. For motor commands to the body wall, a synapse occurs at the chain ganglia, and then the postgangliotic fibers return to the spinal nerve for distribution. For the thoracic cavity, a synapse also occurs at the chain ganglia, but the postgangliotic fibers then form nerves that go directly to their targets. The collateral ganglia. The abdominopelvic tissues and organs receive sympathetic innervation over pregangliotic fibers from lower thoracic and upper lumbar segments that pass through the sympathetic chain without synapsing. Instead, these fibers synapse within 3 unpaired collateral ganglia, figure 8 to 34. The nerves traveling to the collateral ganglia are known as splanchnic nerves. The postgangliotic fibers leave the collateral ganglia and innervate organs throughout the abdominopelvic cavity. The adrenal medulla. Pregangliotic fibers enter each adrenal gland and proceed to its center, the adrenal medulla. The fibers then synapse on modified neurons with an endocrine function. When stimulated, these cells release the neurotransmitters norepinephrine, NE, and epinephrine, E, into the bloodstream. The bloodstream carries NE and E throughout the body, where they cause metabolic changes in many different cells. In general, the effects of these neurotransmitters resemble those produced by the stimulation of sympathetic postgangliotic fibers. General functions of the sympathetic division. The sympathetic division stimulates tissue metabolism, increases alertness, and prepares the individual for sudden, intense physical activity. Sympathetic innervation by the spinal nerve stimulates sweat gland activity and erector pili muscles, producing goosebumps, reduces circulation to the skin and body wall, speeds up blood flow to skeletal muscles, releases stored lipids from adipose tissue, and dilates the pupils. The activation of the sympathetic nerves to the thoracic cavity accelerates the heart rate, increases the force of cardiac contractions, and dilates the respiratory passageways. The postgangliotic fibers from the collateral ganglia reduce the blood flow to visceral organs that are not important to short-term survival, such as the digestive tract. These fibers also reduce energy used by these organs. In addition, the fibers stimulate the release of stored energy reserves. The release of NAND by the adrenal medulla broadens the effects of sympathetic activation to cells not innervated by sympathetic postganglionic fibers. The effects also last much longer than those produced by direct sympathetic innervation. The parasympathetic division. The parasympathetic division of the ANS, figure 8 to 35, includes the following structures. Preganglionic neurons in the brainstem and in sacral segments of the spinal cord. The midbrain, pons, and medulla oblongata contain autonomic nuclei associated with cranial nerves 3, 7, 9, and X. Other autonomic nuclei lie in the lateral gray horns of spinal cord segments S2 to S4. Ganglionic neurons in peripheral ganglia within or adjacent to the target organs. Preganglionic fibers of the parasympathetic division do not diverge as extensively as those of the sympathetic division. For this reason, the effects of parasympathetic stimulation are more localized and specific than those of the sympathetic division. Organization of the parasympathetic division. Figure 835 diagrams the pattern of parasympathetic innervation. Preganglionic fibers leaving the brain travel within cranial nerves 3, oculomotor, 7, facial, 9, glossopharyngeal, and X, vagus. These fibers synapse in terminal ganglia located in peripheral tissues. Short postganglionic fibers then continue to their targets. The biggest nerves provide preganglionic parasympathetic innervation to ganglia in organs of the thoracic and abdominal pelvic cavities. Some of these ganglia are as distant as the last segments of the large intestine. The biggest nerves provide roughly 75% of all parasympathetic outflow, efferent activity, and innervate most of those organs. Preganglionic fibers in the sacral segments of the spinal cord carry the sacral parasympathetic output. They do not join the spinal nerves but instead form pelvic nerves. These nerves innervate intraneural ganglia in the kidney and urinary bladder, the last segments of the large intestine, and the sex organs. General functions of the parasympathetic division. The functions of the parasympathetic division center on relaxation, food processing, and energy absorption. For example, the parasympathetic division constricts the pupils, increases secretions by the digestive glands, increases smooth muscle activity of the digestive tract, stimulates defecation and urination, constricts respiratory passageways, and reduces heart rate and the force of cardiac contractions. This division has been called the anabolic system, anabole, or raising up, because its stimulation leads to a general increase in the nutrient content of the blood. In response, cells throughout the body absorb nutrients and use them in growth, cell division, and the storage of energy reserves. The effects of parasympathetic stimulation are usually brief and are restricted to specific organs and sites. Relationships between the sympathetic and parasympathetic divisions. The sympathetic division has widespread effects, reaching visceral and somatic structures throughout the body. In contrast, the parasympathetic division innervates areas serviced by the cranial nerves and organs in the thoracic and abdominal pelvic cavities. Some organs are innervated by one division or the other, but most vital organs receive dual innervation. That is, they get instructions from both autonomic divisions. In these cases, the two divisions obtain opposing effects. Table 8-2 provides examples of the effects of either single or dual innervation on selected organs. Table 8-2 the effects of the sympathetic and parasympathetic divisions of the ANS on various body structures. Checkpoint. While out for a brisk walk, Megan is suddenly confronted by an angry dog. Which division of the ANS is responsible for the physiological changes that occur as she turns and runs from the animal? Why is the parasympathetic division of the ANS sometimes referred to as the anabolic system? What effect would loss of sympathetic stimulation have on the flow of air into the lungs? What physiological changes would you expect in a patient who is about to undergo a root canal procedure and is quite anxious about it? See the Blue Answers tab at the back of the book. 8 to 12 aging produces various structural and functional changes in the nervous system. Learning outcome. Summarize the effects of aging on the nervous system. Age-related anatomical and physiological changes in the nervous system probably begin by age 30 and accumulate over time. An estimated 85% of individuals above age 65 lead relatively normal lives, but they exhibit noticeable changes in mental performance and in CNS function. Common age-related anatomical changes include a reduction in brain size and weight. This change results primarily from a decrease in the volume of the cerebral cortex. The brains of elderly individuals have narrower gyri and wider sulci than those of young people. A reduction in the number of neurons. Brain shrinkage has been linked to a loss of cortical neurons. Evidence indicates that the loss of neurons does not occur, at least to the same degree, in brainstem nuclei. A decrease in blood flow to the brain. With age, the gradual accumulation of fatty deposits in the walls of blood vessels reduces the rate of blood flow through arteries. This process, called arteriosclerosis, affects arteries throughout the body. We discuss it further in Chapter 13. The reduction in blood flow may not cause a cerebral crisis, but it does increase the chances that the individual will suffer a stroke. Changes in the synaptic organization of the brain. The number of dendritic branchings and interconnections appears to decrease. Synaptic connections are lost, and neurotransmitter production declines. Intracellular and extracellular changes in CNS neurons. Many neurons in the brain accumulate abnormal intracellular deposits, pigments or abnormal proteins, that have no apparent function. Extracellular accumulations of proteins, plaques, may also occur. These changes appear to take place in all aging brains. When present in excess, they seem to be associated with clinical abnormalities. These anatomical changes are linked to impaired neural function. Memory consolidation, the conversion of short-term memory to long-term memory often becomes more difficult. Other memories, especially those of the recent past, also become more difficult to recall. The sensory systems of the elderly, notably hearing, balance, vision, smell, and taste, become less acute. Light must be brighter, sounds louder, and smells stronger before they are perceived. Reaction times are slowed, and reflexes, even some withdrawal reflexes, weaken or disappear. The precision of motor control decreases, so it takes longer to perform a given motor pattern than it did 20 years earlier. For roughly 85% of the elderly population, these changes do not interfere with their abilities to function. But for as yet unknown reasons, some individuals become incapacitated by progressive CNS changes. These changes, which can include memory loss, an inability to recall new information, and emotional disturbances, are often lumped together as dementia. Dementia is a decline in mental ability characterized by deficits in memory, spatial orientation, language, or personality. The most common cause of dementia is Alzheimer's disease. See the clinical note on the next page. Checkpoint. What is the major cause of age-related reduction, or shrinkage, of the brain? See the Blue Answers tab at the back of the book. Clinical note: Alzheimer's disease. Alzheimer's disease is a progressive disorder characterized by the loss of higher cerebral functions. It is the most common cause of dementia in older people. Signs and symptoms may appear at 50 to 60 years of age or later, but the disease occasionally affects younger people. An estimated 5.5 million people in the United States have some form of the condition, including about 16% of those between ages 65 to 74, and 38% over age 85. The disease causes approximately 100,000 deaths each year. It can also have devastating emotional effects on the patient's immediate family. A distressed elderly man rests his head against his hand. In its characteristic form, Alzheimer's disease produces a gradual deterioration of mental organization. The afflicted individual loses memories, verbal and reading skills, and emotional control. As memory losses continue to accumulate, problems become more severe. The affected person may forget relatives, a home address, or how to use the telephone. The loss of memory affects both intellectual and motor abilities. A patient with severe Alzheimer's disease has difficulty performing even the simplest motor tasks. There is no cure for Alzheimer's disease. A few medications and supplements slow its progress in many patients. A spring tied around an index finger. 8 to 13. The nervous system is closely integrated with other body systems. Learning outcome. Give examples of interactions between the nervous system and other body systems presented so far. The nervous system monitors all other systems and issues commands that adjust their activities. Build your knowledge, how the nervous system integrates with the other body systems presented so far on P297 reviews the major functional relationships between it and the integumentary, skeletal, and muscular systems. Checkpoint. Identify the relationships between the nervous system and the other body systems studied so far. See the Who Answers tab at the back of the book. Related clinical. Clinical terms. Amyotrophic lateral sclerosis, ALS. A progressive, degenerative disorder affecting motor. Related clinical terms. Related clinical terms. Amyotrophic lateral sclerosis, ALS. A progressive, degenerative disorder affecting motor neurons of the spinal cord, brainstem, and cerebral hemispheres, commonly known as Lou Gehrig's disease. Cerebrovascular accident, CVA, or stroke. A condition in which the blood supply to a portion of the brain is blocked off. Diphtheria. A disease that results from a bacterial infection of the respiratory tract. Among other effects, the bacteria and toxins damage Schwann cells and cause PN demyelination. Hansen's disease, leprosy. A bacterial infection that begins in sensory nerves of the skin and gradually progresses to a motor. Related clinical terms. Amyotrophic lateral sclerosis, ALS. A progressive, degenerative disorder affecting motor neurons of the spinal cord, brainstem, and cerebral hemispheres, commonly known as Lou Gehrig's disease. Cerebrovascular accident, CVA, or stroke. A condition in which the blood supply to a portion of the brain is blocked off. Diphtheria. A disease that results from a bacterial infection of the respiratory tract. Among other effects, the bacteria and toxins damage Schwann cells and cause PN demyelination. Hansen's disease, leprosy. A bacterial infection that begins in sensory nerves of the skin and gradually progresses to a motor paralysis of the same regions. Huntington's disease. An inherited disease marked by progressive deterioration of mental abilities and by motor disturbances. Meningitis. Inflammation of the meninges involving the spinal cord, spinal meningitis, and / or brain, cerebral meningitis. It is generally caused by bacterial or viral pathogens. Myelography. A diagnostic procedure in which radiopig dye is introduced into the cerebrospinal fluid to obtain an X-ray image of the spinal cord and cauda equina. Neurology. The branch of medicine that deals with the study of the nervous system and its disorders. Neurotoxin. A compound that disrupts normal nervous system function by interfering with the generation or propagation of action potentials. Examples include tetrodotoxin, TTX, saxitoxin, STX, paralytic shellfish poisoning, PSP, and sigmodoxin, CTX. Paresthesia. An abnormal tingling sensation, usually described as pins and needles, that accompanies the return of sensation after a temporary palsy. Sciatica, sciatica. The painful result of compression of the roots of the sciatic nerve. Shingles. A condition caused by the infection of neurons in spinal ganglia by the varicella zoster virus. The primary symptom is a painful rash along the sensory distribution of the affected spinal nerves. Spinal shock. A period of depressed sensory and motor function following any severe injury to the spinal cord. Build your knowledge. How the nervous system integrates with the other body systems presented so far. Text discusses how the nervous system interacts with the integumentary, skeletal, and muscular systems. A chapter review. Summary outline. An introduction to the nervous system. B. 244. Two organ systems, the nervous and endocrine systems, coordinate organ system activity. The nervous system provides swift but brief responses to stimuli. The endocrine system adjusts metabolic operations and directs long-term changes. 8 to 1. The nervous system has anatomical and functional divisions. B. 244. The nervous system includes all the nervous tissue in the body. Its major anatomical divisions include the central nervous system, CNS, the brain and spinal cord, and the peripheral nervous system, PNs, all the nervous tissue outside the CNS. Functionally, the PNs can be divided into an afferent division, which brings sensory information to the CNS, and an efferent division, which carries motor commands to muscles and glands. The efferent division includes the somatic nervous system, SNS, voluntary control over skeletal muscle contractions, and the autonomic nervous system, ANS, automatic, involuntary regulation of smooth muscle, cardiac muscle, glandular activity, and adipose tissue. Figure 8 to 1. 8 to 2 neurons are specialized for intercellular communication and are supported by cells called neuroglial. 245. There are two types of cells in nervous tissue, neurons, which are responsible for information transfer and processing, and neuroglial cells, which regulate the environment around neurons, provide a supporting framework, and act as phagocytes. Sensory neurons form the afferent division of the PNs and deliver information to the CNS. Motor neurons stimulate or modify the activity of the peripheral tissue, organ, or organ system. Interneurons, association neurons, may be located between sensory and motor neurons. They analyze sensory inputs and coordinate motor outputs. A typical neuron has a cell body, an axon, several branching dendrites, and axon terminals. Figure 8 to 2. Neurons may be described as unipolar, bipolar, or multipolar. Figure 8 to 3. The four types of neuroglia in the CNS are 1. Astrocytes, which are the largest and most numerous and maintain the blood-brain barrier. 2. Oligodendrocytes, which are responsible for the myelination of CNS axons. 3. Microglia, phagocytic cells derived from white blood cells. And 4. Ependymal cells, with functions related to the cerebrospinal fluid, CSF. Figure 8 to 4. Nerve cell bodies in the PNs are clustered into ganglia, singular, ganglion. Their axons are covered by myelin wrappings of Schwann cells. Figure 8 to 5. In the CNS, a collection of neuron cell bodies that share a particular function is called a center. A center with a discrete anatomical boundary is called the nucleus. Areas of the brain surface are covered by a thick layer of gray matter called the neural cortex. The white matter of the CNS contains bundles of axons, or tracts, that share common origins, destinations, and functions. Tracts in the spinal cord form larger groups, called columns. Figure 8 to 6. Sensory, ascending. Pathways carry information from peripheral sensory receptors to processing centers in the brain. Motor, descending. Pathways extend from CNS centers concerned with motor control to the associated skeletal muscles. Figure 8 to 6. 8 to 3 in neurons. A change in the plasma membrane's electrical potential may result in an action potential, nerve impulse. B. 251. The resting membrane potential of an undisturbed nerve cell results from a balance between the rates of sodium ion gain and potassium ion loss achieved by the sodium-potassium exchange pump. Any stimulus that affects this balance will alter the resting potential of the cell. Figure 8 to 7. An action potential appears when the membrane depolarizes to a level known as the threshold. The steps involved include depolarization to threshold, the opening of voltage-gated sodium ion channels and membrane depolarization, the closing of voltage-gated sodium ion channels and opening of voltage-gated potassium ion channels, and the return to normal permeability. Spotlight figure 8 to 8. In continuous propagation, an action potential spreads along the entire excitable membrane surface in a series of small steps. During saltatory propagation, the action potential appears to lead from node to node, skipping the intervening myelinated membrane surface. Spotlight figure 8 to 9. 8 to 4 at synapses. Communication takes place among neurons or between neurons and other cells. B. 258. A synapse is a site where a neuron communicates with another cell through the release of chemicals called neurotransmitters. A synapse where neurons communicate with other cell types is a neurovector junction. Neural communication moves from the presynaptic neuron to the postsynaptic neuron across the synaptic cleft. Figure 8 to 10. Cholinergic synapses release the neurotransmitter acetylcholine, ACH. ACH is broken down in the synaptic cleft by the enzyme acetylcholinesterase, ACE. Figure 8 to 11. The roughly 20 billion interneurons are organized into neuronal pools, groups of interconnected neurons with specific functions. Divergence is the spread of information from one neuron to several neurons or from one neuronal pool to several pools. In convergence, several neurons synapse on the same postsynaptic neuron. Figure 8 to 12. 8 to 5. The brain and spinal cord are surrounded by 3 layers of membranes called the meninges. B. 261. The CNS is made up of the spinal cord and brain. Special covering membranes, the meninges, protect and support the spinal cord and the delicate brain. The cranial meninges, dura mater, arachnoid, and via mater, are continuous with those of the spinal cord, the spinal meninges. Figure 8 to 13. The dura mater covers the brain and spinal cord. The epidural space separates the spinal dura mater from the walls of the vertebral canal. The subarachnoid space of the arachnoid layer contains cerebrospinal fluid, the CSF, which acts as a shock absorber and a diffusion medium for dissolved gases, nutrients, chemical messengers, and waste products. The pia mater is bound to the underlying neural tissue. 8 to 6. The spinal cord contains gray matter surrounded by white matter and connects to 31 pairs of spinal nerves. B. 262. In addition to relaying information to and from the brain, the spinal cord integrates and processes information on its own. The spinal cord has 31 segments, each associated with a pair of spinal ganglia and their posterior roots, and a pair of anterior roots. Figures 8 to 14, 8-15b. The white matter contains myelinated and unmyelinated axons. The gray matter contains cell bodies of neurons and glial cells. The projections of gray matter toward the outer surface of the spinal cord are called horns. Figure 8-15a. 8 to 7 the brain has several principal structures, each with specific functions. 266. There are 6 regions in the adult brain: cerebrum, diencephalon, midbrain, pons, medulla oblongata, and cerebellum. Figure 8 to 16. The central passageway of the brain expands to form 4 chambers called ventricles. Cerebrospinal fluid, CFS, continuously circulates from the ventricles and central canal of the spinal cord into the subarachnoid space of the meninges that surround the CNS. Figures 8 to 17, 8 to 18. Conscious thought, intellectual functions, memory, and complex involuntary motor patterns originate in the cerebrum. Figure 8 to 16. The cortical surface of the cerebrum contains gyri, elevated ridges, separated by sulci, shallow depressions, or deeper grooves, fissures. The longitudinal fissure separates the 2 cerebral hemispheres. The central sulcus marks the boundary between the frontal lobe and the parietal lobe. Other sulci form the boundaries of the temporal lobe and the occipital lobe. Figures 8 to 16, 8 to 19. Each cerebral hemisphere receives sensory information and generates motor commands that concern the opposite side of the body. The primary motor cortex of the percentral gyrus directs voluntary movements. The primary somatosensory cortex of the postcentral gyrus receives somatic sensory information from touch, pressure, pain, and temperature receptors. Association areas, such as the visual association area and premotor cortex, somatic motor association area, control our ability to understand sensory information and coordinate a motor response. Figure 8 to 19. The left hemisphere is usually the categorical hemisphere, which contains the Wernicke's area and Broca's area, speech center, and is responsible for language-based skills. The right hemisphere, or representational hemisphere, is concerned with spatial relationships and analyses. Figure 8 to 20. An electroencephalogram, EEG, is a printed record of brain waves. Figure 8 to 21. The basal nuclei lie within the central white matter and aid in the coordination of learned movement patterns and other somatic motor activities. Figure 8 to 22. The limbic system is a functional grouping rather than an anatomical one. Among other structures, it includes the hippocampus, which is involved in memory and learning, and the mammillary bodies, which control reflex movements associated with eating. The functions of the limbic system involve establishing emotional states, linking the conscious, intellectual functions of the cerebral cortex with the unconscious and autonomic functions of the brainstem, and aiding long-term memory storage and retrieval. Figure 8 to 23. The diencephalon provides the switching and relay centers needed to integrate the conscious and unconscious sensory and motor pathways. It is made up of the epithalamus, which contains a pineal gland and choroid plexus, a vascular network that produces cerebrospinal fluid, the thalamus, and the hypothalamus. Figures 8-16c, 8 to 24. The thalamus is the final relay point for sending sensory information. Only a small portion of the arriving sensory information is passed to the cerebral cortex. The rest is relayed to the basal nuclei and centers in the brainstem. Figures 8-16c, 8 to 24. The hypothalamus contains important control and integrative centers. It can produce emotions and behavioral drives, coordinate activities of the nervous and endocrine systems, secrete hormones, coordinate voluntary and autonomic functions, and regulate body temperature. 3 regions make up the brainstem. 1. The midbrain processes visual and auditory information and generates involuntary somatic motor responses, and contains a reticular activating system, RAS, which directly affects the activity of the cerebral cortex. 2. The pons connects the cerebellum to the brainstem and is involved with somatic and visceral motor control. 3. The spinal cord connects to the brain at the medulla oblongata, which relays sensory information and regulates autonomic functions. Figures 8-16c, 8 to 24. The cerebellum oversees the body's postural muscles and programs and fine-tunes voluntary and involuntary movements. The cerebellar peduncles are tracts linking the cerebellum with the brainstem, cerebrum, and spinal cord. Figures 8 to 16, 8 to 24. The medulla oblongata connects the brain to the spinal cord. Its nuclei relay information from the spinal cord and brainstem to the cerebral cortex. Its reflex centers, including the cardiovascular center and the respiratory rhythmicity centers, control or adjust the activities of one or more peripheral systems. Figure 8 to 24. 8 to 8 the PN connects the CNS with the body's external and internal environments. 278. The peripheral nervous system, PNs, links the central nervous system, CNS, with the rest of the body. All sensory information and motor commands are carried by axons of the PNs. The sensory and motor axons are bundled together into peripheral nerves, or nerves, and clusters of cell bodies, or ganglia. The PNs includes cranial nerves and spinal nerves. There are 12 pairs of cranial nerves, which connect to the brain, not to the spinal cord. Figure 8 to 25. The olfactory nerves, carry sensory information for the sense of smell. The optic nerves, 2, carry visual information from special sensory receptors in the eyes. The oculomotor nerves, 3, are the primary sources of innervation for 4 of the 6 muscles that move the eyeball. The trochlear nerves, 4, the smallest cranial nerves, innervate the superior oblique muscles of the eyes. The trigeminal nerves, the largest cranial nerves, are mixed nerves with 3 divisions, the ophthalmic nerve, the maxillary nerve, and the mandibular nerve. The epicent nerves, 6, innervate the 6th extrinsic eye muscle, the lateral rectus. The facial nerves, 7, are mixed nerves that control muscles of the scalp and face. They provide pressure sensations over the face and receive taste information from the tongue. The vestibular cochlear nerves, 8, contain the vestibular nerves, which monitor sensations of balance, position, and movement, and the cochlear nerves, which monitor hearing receptors. The glossopharyngeal nerves, 9, are mixed nerves that innervate the tongue and pharynx and control swallowing. The vagus nerves, X, are mixed nerves that are vital to the autonomic control of visceral function and have a variety of motor components. The accessory nerves, 11, have an internal branch, which innervates voluntary swallowing muscles of the soft palate and pharynx, and an external branch, which controls muscles associated with the pectoral girdles. The hypoglossal nerves, 12, provide voluntary control over tongue movements. There are 31 pairs of spinal nerves, 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and 1 coccygeal. Each pair monitors a region of the body surface known as a dermatome. Figures 8 to 26, 8 to 27. A nerve plexus is a complex, interwoven network of nerves. The 4 large plexuses are the cervical plexus, the brachial plexus, the lumbar plexus, and the sacral plexus. The latter 2 can be united into a lumbosacral plexus. Figure 8 to 26. 8 to 9 reflexes are rapid, automatic responses to stimuli. 283. A reflex is an automatic involuntary motor response to a specific stimulus. A reflex arc is the wiring of a single reflex. 5 steps are involved in the action of a reflex arc. 1. Arrival of a stimulus and activation of a receptor. 2. Activation of a sensory neuron. 3. Information processing. 4. Activation of a motor neuron. And 5. Response by an effector. Figure 8 to 28. A monosynaptic reflex is the simplest reflex arc, in which a sensory neuron synapses directly on a motor neuron that acts as the processing center. A stretch reflex is a monosynaptic reflex that automatically regulates skeletal muscle length and muscle tone. The sensory receptors involved are Figure 8 to 29. Polysynaptic reflexes, which have at least one interneuron between the sensory afferent neuron and the motor afferent neuron, have a longer delay between stimulus and response than does a monosynaptic synapse. Polysynaptic reflexes can also produce more complex responses. A flexor reflex is a withdrawal reflex affecting the muscles of a limb. Figure 8 to 30. The brain can facilitate or inhibit reflex motor patterns based in the spinal cord. 8 to 10 separate pathways carry sensory information and motor commands. 286. The essential communication between the CNS and PNs occurs over pathways that relay sensory information and motor commands. Table 8 to 1. A sensation arrives in the form of an action potential in an afferent fiber. The posterior column pathway carries fine touch, pressure, vibration, and proprioceptive sensations. The axons ascend within this pathway and synapse with neurons in the medulla oblongata. These axons then cross over and travel onto the thalamus. The thalamus sorts the sensations according to the region of the body involved and sends them to specific regions of the primary somatosensory cortex. Figure 8 to 31. Table 8 to 1. The corticospinal pathway provides conscious skeletal muscle control. The medial and lateral pathways generally exert subconscious control over skeletal muscles. Figure 8 to 32. Table 8 to 1. 8 to 11. The autonomic nervous system, composed of the sympathetic and parasympathetic divisions, is involved in the unconscious regulation of body functions. 289. The autonomic nervous system, ANS, coordinates cardiovascular, respiratory, digestive, excretory, and reproductive functions. Preganglionic neurons in the CNS and axons to synapse on ganglionic neurons in autonomic ganglia outside the CNS. The axons of the ganglionic neurons, postganglionic fibers, innervate cardiac muscle, smooth muscles, glands, and, in some cases, fat cells. Figure 833b. Preganglionic fibers from the thoracic and lumbar segments form the sympathetic division. Fight or flight system of the ANS. Preganglionic fibers leaving the brain and sacral segments form the parasympathetic division. Rest and repose or rest and digest system. The sympathetic division consists of preganglionic neurons between spinal segments T1 and L2, ganglionic neurons in ganglia near the vertebral column, and specialized neurons in the adrenal gland. Sympathetic ganglia are paired sympathetic chain ganglia or unpaired collateral ganglia. Figure 8 to 34. Preganglionic fibers entering the adrenal gland synapse within the adrenal medulla. During sympathetic activation, these endocrine organs secrete epinephrine, E, and norepinephrine, NA, into the bloodstream. In a crisis, the entire sympathetic division responds, producing increased alertness, a feeling of energy, increased cardiovascular and respiratory activity, and elevation in muscle tone. The parasympathetic division includes preganglionic neurons in the brainstem and sacral segments of the spinal cord, and ganglionic neurons in peripheral ganglia located within or next to target organs. Preganglionic fibers leaving the sacral segments form pelvic nerves. Figure 8 to 35. The effects produced by the parasympathetic division center on relaxation, food processing, and energy absorption. They are usually brief and restricted to specific sites. The sympathetic division has widespread effects, reaching visceral and somatic structures throughout the body. The parasympathetic division innervates only visceral structures either serviced by cranial nerves or lying within the abdominopelvic cavity. Organs with dual innervation receive instructions from both divisions. Table 8 to 2. 8 to 12 aging produces various structural and functional changes in the nervous system. 294. Age-related changes in the nervous system include a reduction of brain size and weight, a reduction in the number of neurons, decreased blood flow to the brain, changes in synaptic organization of the brain, and intracellular and extracellular changes in CNS. For roughly 85% of the elderly population, these changes do not interfere with their abilities to function. 8 to 13. The nervous system is closely integrated with other body systems. 296. The nervous system monitors all other body systems and issues commands that adjust their activities. Review questions. See the Blue Answers tab at the back of the book. Level 1 reviewing facts and terms. Match each item in column A with the most closely related item in column B. Place letters for answers in the spaces provided. Column A. 1 neural gland. 2 autonomic nervous system. 3 sensory neurons. 4 dual innervation. 5 ganglia. 6 oligodendrocytes. 7 ascending tracts. 8 descending tracts. 9 solitary propagation. 10 continuous propagation. 11 duromotor. 12 monosynaptic reflex. 13 sympathetic division. 14 cerebellum. 15 somatic nervous system. 16 hypothalamus. 17 medulla oblongata. 18 choroid plexus. 19 parasympathetic division. 20 motor neurons. Column B. Column A. 1. 19 parasympathetic division. 20 motor neurons. Column B. Fight or flight division. 21 label the structures in the following diagram of a neuron. Identify the structures in the diagram of a neuron. 22 regulation by the nervous system provides relatively slow but long-lasting responses to stimuli. Swift, long-lasting responses to stimuli. Swift but brief responses to stimuli. Relatively slow, short-lived responses to stimuli. 23 all the motor neurons that control skeletal muscles are multipolar neurons. Myelinated bipolar neurons. Unipolar. Unmyelinated sensory neurons. Proprioceptors. 24 depolarization of a neuron plasma membrane will shift the membrane potential toward 0 MV. 70 MV. 90 MV. A, B, and C are correct. 25 identify the 6 principal regions of the brain in the following diagram. Identify the regions in the brain, brainstem and spinal cord. 26 the structural and functional link between the cerebral hemispheres and the components of the brainstem is the neural cortex. Medulla oblongata. Midbrain. Diencephalon. 27 the ventricles in the brain are filled with blood. Cerebrospinal fluid. Air. Nervous tissue. 28 reading, writing, and speaking are dependent on processing in the right cerebral hemisphere. Left cerebral hemisphere. Prefrontal cortex. Post central gyrus. 29 establishment of emotional states and related behavioral drives are functions of the limbic system. Pineal gland. Mammillary bodies. Thalamus. 30 the final relay point for sending sensory information that will be projected to the primary somatosensory cortex is the hypothalamus. Thalamus. Spinal cord. Medulla oblongata. 31 spinal nerves are called mixed nerves because they are associated with a pair of spinal ganglia. Exit and intervertebral foramina. Contain sensory and motor fibers. Are associated with a pair of dorsal and ventral roots. 32 there is always a synapse between the CNS and the peripheral effector in the ANS. The SNS. A reflex arc. A, B, and C are correct. 33 approximately 75% of parasympathetic efferent activity is provided by the pelvic nerves. Sciatic nerves. Glossopharyngeal nerves. Vagus nerves. 34 state the all-or-none principle of action potentials. 35 using the mnemonic device O. Once one takes the anatomy final, very good vacations are heavenly. Listed 12 pairs of cranial nerves. 36 how does the emergence of sympathetic preganglionic fibers from the spinal cord differ from the emergence of parasympathetic preganglionic fibers? Level 2 reviewing concepts. 37 A graded potential. Decreases with distance from the point of stimulation. Spreads passively because of local currents. May involve either depolarization or hyperpolarization. A, B, and C are correct. 38 the loss of positive ions from the interior of a neuron produces depolarization. Threshold. Hyperpolarization. And action potential. 39 which major part of the brain is associated with respiratory and cardiac activity. 40 why is response time in a monosynaptic reflex much faster than response time in a polysynaptic reflex? 41 compare the general effects of the sympathetic and parasympathetic divisions of the ANS on mental alertness, metabolic rate, digestive/urinary function, use of energy reserves, respiratory rate, heart rate/blood pressure, and sweat gland activity. Level 3 critical thinking and clinical applications. 42 of neurons in the central nervous system lack centrioles and are unable to divide. How can a person develop brain cancer? 43 a police officer has just stopped Bill on suspicion of driving while intoxicated. The officer asked Bill to walk the yellow line on the road and then asked him to place the tip of his index finger on the tip of his nose. Which part of the brain is being tested by these activities? How would these activities indicate Bill's level of sobriety? 44 in some severe cases of stomach ulcers, the branches of the vagus nerve, S, that lead to the stomach are surgically severed. How might this procedure control the ulcers? 45 improper use of crutches can produce a condition known as crush paralysis, which is characterized by a lack of response by the extensor muscles of the arm and a condition known as wrist drop. Which nerve is involved? 46 while playing football, Ramon is tackled hard and suffers an injury to his left leg. As he tries to get up, he finds that he cannot flex his left hip or extend the knee. Which nerve is damaged, and how would this damage affect sensory perception in his left leg? Mastering A and B. Access more study tools online in the study area of Mastering A and B. Chapter pretest. Chapter quizzes. Chapter practice test. Animations. Practice anatomy lab trademark, PAL trademark, 3.1 virtual anatomy practice tool. Interactive physiology registered, IP, animations. MP3 tutor sessions. Flashcards and glossary. For this chapter, follow these navigation paths in PAL. Human cadaver nervous system. Anatomical models nervous system. Histology nervous tissue. For this chapter, go to these topics in the nervous system in IP. Resting membrane potential. Generation of an action potential. Propagation and velocity of the action potential. Events at the synapse. For this chapter, go to these topics in the MP3 tutor sessions. Generation of an action potential. Differences between the sympathetic and parasympathetic nervous systems. 9.